Once everything's perfectly organized. Awesome.
Sir, NRC తనకు audible sir voice. NRC DAW GDC Nelluru audible madam.
గుడ్ మార్నింగ్ ఆల్. ఒకసారి మీ ఆడియో టెస్ట్ చేసుకోండి ఒకసారి. ఉమెన్ శ్రీకాకుళం. ఉమెన్ శ్రీకాకుళం, ప్లీజ్ రెస్పాండ్. సార్ ఉమెన్ శ్రీకాకుళం కొంచెం కంటిన్యూ మాట్లాడండి, ఆడియో కొంచెం బ్రేక్ అవుతున్నట్టుంది. ఎక్కువ వస్తుంది సార్, ఒకసారి ట్రై చేయండి. మళ్ళీ ఉమెన్ శ్రీకాకుళం.
రెస్పాండింగ్ సార్.
సార్ ఎక్కువ వస్తుంది సార్, చూసుకోండి. ఉమెన్ విశాఖపట్నం.
Sir, responding sir. Good morning sir.
అంటే వీళ్ళు
Responding.
ఓకే మేడం. తణుకు ప్లీజ్ రెస్పాండ్.
Yes, sir. Good morning, sir.
గుడ్ మార్నింగ్ సార్.
Sir, our audio is clear, sir. Rambam,
ఎస్ సార్, క్లియర్ సార్,
Yes, sir.
పర్ఫెక్ట్.
Thank you.
కాకినాడ ప్లీజ్ రెస్పాండ్.
Sir, NRC backline response.
ఓకే సార్. గుంటూరు ప్లీజ్ రెస్పాండ్.
Sir, good morning, sir.
గుడ్ మార్నింగ్ సార్.
Good morning.
Am I audible, sir?
ఆడిబుల్ సార్.
Audible,
Yes.
sir?
Yes. Can you
Okay,
hear
sir.
me?
Thank you, sir.
ఎస్ సార్. సార్ యు ఆర్ పర్ఫెక్ట్ ఆల్రెడీ సార్. భట్టాచార్య సార్.
Right. Right.
So 5 minutes, sir.
Sure. No problem.
DKW Nellore, please respond.
Responding, sir.
Madam, కొంచెం వాల్యూమ్ ఇంక్రీజ్ చేసుకోండి.
Nellore responding, sir.
ఓకే మ్యామ్. చిత్తూరు ప్లీజ్ రెస్పాండ్. PVKN చిత్తూరు. PVKN చిత్తూరు ప్లీజ్ రెస్పాండ్. PVKN చిత్తూరు ప్లీజ్ రెస్పాండ్.
Hello, good morning, sir. Hello. Sir, good morning, sir.
సార్ కంటిన్యూ మాట్లాడండి.
Chitur responding, sir. NRC PVKN responding, sir.
సార్ ఓకే, కానీ ఏంటంటే మధ్య మధ్యలో కొంచెం డిస్టర్బ్ ఉన్నట్టుంది, ఒకసారి చెక్ చేసుకోండి ఆడియో.
Okay.
అనంతపూర్ ప్లీజ్ రెస్పాండ్. Anantapur, please respond.
Sir, GDC Min Anantpur is responding.
Yeah, okay, Nandi.
Video clear, sir?
Clear, sir.
Audio, video.
NRC Kadapa. NRC Kadapa, please respond. NRC Kadapa, please respond. Kadapa, please respond. NRC Kadapa. NRC Kadapa, please respond. Already the resource person has joined.
Should we start?
Sir, just a minute, sir.
Okay, okay.
సార్ నమస్తే సార్ పడకండి సార్
Madam, namaste, madam. Meer Kadapa nuncha, madam.
హలో
Hello.
హలో
Hello. Varalakshmi, madam.
హలో
Madam, you are not audible.
హలో
Madam, వినిపిస్తుంది. మీరు
Hello?
సార్ని ఇంటర్వ్యూ చేసేయండి.
హలో సార్ Hello.
Hello.
Sorry, my voice is audible.
Yes, madam. One question, madam. Is audible, madam?
Hello.
One question, madam. You going to introduce research person?
Sir Srinivas sir, I think audio problem is there, sir, from Varalakshmi madam side. So you please introduce our today's resource person, sir.
True, sir. True. We have all the NRCs of Andhra Pradesh. I deem it a privilege to introduce one of the renowned scientists from University of Hyderabad, that is Professor Bhattacharya Pranalakanti. He completed his BSc Chemistry in 1990 from Ramakrishna Mission, Vivekananda Centenary College, University of Calcutta, and MSc Biochemistry in 1992 from University of Calcutta. He performed his PhD work for his molecular biology between 1993 and 1998 at Tata Institute of Fundamental Research, Mumbai, where he investigated molecular aspects of naturally acquired immunity to human malarial parasite, that is Plasmodium falciparum. Professor Bhattacharya worked as postdoctoral research fellow at John Hopkins University, USA from 1999 to 2004, where he initiated a study on homologous recombination, that is HR, with Plasmodium falciparum, and he began to understand how HR should be involved in the generation of genomic diversity parasites. He started his career as a research scientist in Tulane University's School of Medicine USA from 2004 to 2007, where he worked on DNA repeat and telomere maintenance of these chromosomes and found intriguing relationship between the MRE11, a protein, the DNA involved in DNA repair, mediated DNA damage response, DDR, and telomere maintenance. He also worked as visiting research scientist in Northwestern University, USA, from October 2005 to January 2006. Dr. Bhattacharya joined University of Hyderabad in March 2007 as the leader of biochemistry. Currently, that is since April 2015, he is a professor of biochemistry. He has served as the head of the Department of Biochemistry from 1st February 2017 to January 31st January 2020. His teaching activities include molecular biology, gene regulation, and recombinant DNA technology. I invite Professor Nodal Bhattacharya to give his presentation on DNA replication. Over to Professor Bhattacharya, sir.
Am I audible to all of you?
Yes, sir. Very much audible, sir.
Great.
Proceed, sir.
Thank you for the detailed and great introduction. So I welcome you all to this session, which will last probably for 1 hour 15 minutes. And so today I'm going to talk on DNA replication. However, today I'm not going to teach on DNA replication because all of you know about DNA replication, what are the mechanisms, what are the enzymes involved and all. Instead, what I will do today is that keeping DNA replication as a topic of discussions, we'll see that how to teach DNA replication for at the postgraduate level. So let me share my screen. Is my screen visible to all of you?
Visible, sir. Visible, sir. Yes, sir. It is visible.
Yes. Oh, great, great. So I think that all DNA replication lecture should start with an experiment that all of you know, which is Michelson-Stahl experiment. Now, in 1950, the DNA structure was solved. And then their model basically suggested how DNA replication can take place. So their model was based on the sequence complementarity, and the prediction of that model was that DNA can be synthesized in a semi-conservative way, where one strand will act as a template, and using that template a new daughter strand can be synthesized. Similarly, the Crick strand can act as a template, and using that another new daughter strand can be synthesized. So that is the semi-conservative mode of replication, which was a part of the model that Watson and Crick proposed. Now, at the same time, people started understanding the complexity of the genome, particularly the limited space in the eukaryotic nucleus, where all these chromosomes are there. Now, Watson-Crick's model suggests that the DNA should be unwound before the replication takes place. Now, how that unwinding can take place within that limited space of the nucleus remained a puzzle. At that time, another famous scientist, Delbruck, he suggested a model that is called breakage and reunion, where basically one DNA strand can be broken, and then through that another you can pass the another DNA strand, thereby you can relieve the strain which is created due to DNA unwinding. And finally, that broken DNA strand can be rejoined or ligated. Now we know that exactly the similar kind of thing happens, and the enzyme involved is topoisomerase, which can cut the DNA, relieve the strain, and then it can join back the DNA, right? But what Delbruck did, that he overcomplicated that model by saying that after DNA break, like after the DNA break happens, then there will be a replication. And finally, downstream somewhere, there will be a ligation to join. So that was his model. And this model is called dispersive model, where both the strands will have the parental old nucleotides and the new nucleotides from the daughter, right? So that was a dispersive. Another third model was proposed, which was purely a theoretical model, and people did not have any idea how that can be achieved or which mechanism can do that. That was the conservative model, where it tells that you have the parental DNA, and from there you synthesize the daughter DNA, and both strands of the daughter DNA will be newly synthesized. Now, how that will be achieved, no idea on that. But those are the three existing model, and there are many scientists, including Watson and Crick, Martin O'Malley, and many other scientists. They wanted to test this model, that how DNA replication is taking place. Only Michelson and Stahl was successful in that, because they discovered a method of separating out the newly synthesized daughter strand from the parental strand. So basically what they did, they used heavy isotope of nitrogen. So once you use heavy isotope of nitrogen, your daughter strand or the newly synthesized strand can be separated out on density gradient centrifugation, right? Martin O'Malley or Watson, Crick and all, they were using radioactivity. Two different radioisotopes, which are not heavy or light isotopes, right? So there they could not physically separate them. So that was the problem. Now, what they did, they grew E. coli cultured in N15 ammonium chloride medium for quite some generations. So now the E. coli genome is N15. Then they transferred it to N14 ammonium chloride medium and grew it for 20 minutes, which is one generation time, and 40 minutes, which is 2 generation time. Now, after 20 minutes, they have isolated the DNA, ran it on a cesium chloride density gradient, and tried to observe that what kind of DNA they are getting. Now, let's look at the expected outcome. What do you expect from such experiment? If it is conservative mode of replication, then the parental DNA will have only N15 ammonium, it will come from only N15, right? And the daughter DNA will be N14. If it is dispersive, then the parental DNA will be a mixture of N14, N15. If it is dispersive, then both the DNA will be a mixture of N14, N15. And if it's semi-conservative, then also in both the DNA, one strand will be N14 and another strand will be N15. So basically it will be a hybrid density. So if it is conservative, it will give you 2 bands, one for N14, N14, one for N15, N15. If it is dispersive or semi-conservative, it will give you 1 band of hybrid density, which is N14, N15. And what they observed was that it is N14, N15. So clearly conservative mode of replication is not taking place. It is either dispersive or semi-conservative, right? Now, how to distinguish between these two? You have to wait for the next cycle. In the next cycle, what will happen? If it is dispersive, then each strand will contain both N14 and N15. So it will be giving hybrid density. But if it is semi-conservative, then you will find 2 DNA strands out of the 4 DNA strands, 2 DNA strands will be, 2 DNA molecules will be N14, N15, and 2 DNA molecules will be N14, N14. So if you run them on a density gradient, if it is dispersive, you will still get a single band of hybrid density. If it is semi-conservative, then you'll get 2 bands, 1 band of hybrid density and another band which is only N14, N14. And that's what was observed. So which tells that the DNA replicate via the semi-conservative mode of replication, and which is actually proposed by Watson and Crick. Now the implication of this experiment is it not only tells that how DNA is replicated, but it actually subsided all those speculations or skepticism that whether DNA can be replicated at all because of the small space in the nucleus and all. Because experimentally you can see that DNA can be replicated via Watson-Crick model, right? So then lots of research started happening in this field, and now instead of solving the topology problem, people are more interested in finding out what are the enzymes that does DNA replication. What are the different enzymes which are involved in many steps of DNA replication? Those are the main thing which people are researching on. So the DNA topology problem was solved after 30 years in 1980s. that topoisomerase can solve the DNA topology problem. But until then, all the DNA replication enzymes were discovered. So now at this point, I would suggest you that you probably give your student a problem on Michelson-Stall experiment. That way, they will understand the intricacy of this experiment much better. So the problem could be like this. Consider Michelson-Stall experiment. So E. coli grown in N15 ammonium chloride containing medium is shifted to N14 ammonium chloride containing medium. Also assume that genomic DNA is fragmented at least in 100 pieces during isolation. Then you have to fill in the table below. So this is important to know that when you isolate DNA, genomic DNA from E. coli, for example, you do not get that unbroken entire chromosome. You get fragmented chromosomes, right? It is fragmented in many different parts, and which is very important to solve this problem, because if it is one single unbroken DNA molecule, that large DNA molecule, this experimental finding would not hold true. So at 0th generation, when you have just transferred it from N15 to N14, all DNA is N15, 100% DNA is N15, because replication did not begin that time. So N14, N15 is 0%, N15 and N14 is also 0%. Now what happens after half generation? So that is very important to understand, that after half generation, what will happen? So let me see. Yeah. So if you can draw the DNA like this, so after half generation, let's assume that half of the chromosome is duplicated, which is this half of the chromosome is duplicated, the other half is not duplicated, because you have is half generation. Now, if you start with N15, so this strand is N15. That strand is also N15. Now this newly synthesized strand, these are N14, right? So now, since the DNA is fragmented, you can imagine that there will be one fragment like this. So this is one fragment. Another, and then there is a third one, right? So this top one, this will be N15, N15. Here, one of them will be N15, another will be N14. So it's a hybrid density, N14, N15. Similarly, this one will also be N14, N15. So what we'll get here is, after half generation, 33% will be N15, N15, and 66% will be N14, N15. And there will be no N14, N14 as of now, right? So this concept is very important to understand. Then you can see that after one generation, what will happen, which is already there in the Michelson-Stall experiment. After one half generation, what will happen, and all those things, right? So this is a very important concept that you can ask your student to solve. Now let's begin on DNA replication. So it's basically genome duplication. In a simplistic term, you have one DNA molecule, from there you get two DNA molecules. And you need some enzymes to do that. And that enzyme is DNA polymerase that you can talk about. And basically what is happening is that you are basically joining the DNTPs in this reaction, right? So at this point, it is very important to understand that what is the substrate of DNA replication. Because if you ask, many students will tell substrate is DNA. And if you ask them what is the product, they'll tell the product is also DNA. Now it is an enzymatic reaction. The substrate and product cannot be the same, right? So the substrates are the DNTPs. And the product is the polymer of that. Right? That's why the enzyme is called polymerase, which is polymerizing DNTPs. So in true sense, the substrates are the DNTPs and also the RNA primer that you know. And the product is the newly synthesized DNA, right? Now, another very important chemical concept at this point is entropy. What is happening? So we know that entropy is the measure of disorderness. If you have all the DNTPs, the dis is very high. If you put it on a chain, like you like it, join one after another. So they make a chain, right? Then the disorderness decreases, right? So if you look at the Gibbs equation, del G is equal to del H minus T del S. You know that for all spontaneous reaction, del G should be negative. Now here, since entropy is decreasing, del G is becoming positive. Right? From DNTPs, you join them together, disorderness is less, entropy decreases, del G increases because del G is equal to del H minus T del S. Right? So if del G is increasing, then how this reaction is possible? This is possible because this is a concept of coupled reaction. Another reaction is coupled with that, which is the hydrolysis of PPI when I release, right? And hydrolysis of PPI takes place, and that has a huge negative delta G. So together, the resultant delta G of this reaction, the two-step reaction, one is addition of DNTPs and another is the hydrolysis of PPI. Together, the delta G is highly negative, and that's how this reaction is possible. So this is a very important concept that the student should understand. Okay. Now there are certain unique features of replication. Replication does not start randomly. It starts at a particular point. And this point is called origin of replication. For circular bacterial chromosome, there is a particular point where it starts and it is called origin of replication. For eukaryotic linear chromosomes, there are multiple origin of replications or ARS. And in yeast, this is a very great model system where a lot of work was done. In yeast, there are about 300 origin of replications, and the distance between each of them is approximately 40 kb. In human, there are about 20,000 origin of replications, and the distance between them are 150 kb. Another interesting concept is replication is bidirectional. The replication fork goes in both directions. So here it is important to understand that all replication goes from 5' to 3' direction. And one strand is copied as a leading strand and another is copied as a lagging strand. We will come to that why such. So basically what happens, that suppose this is copied like that, 5' to 3', and here So this will be the lagging strand. Similarly, this is the leading strand. That is a lagging strand. So which basically tells that suppose you the nomenclature is the top strand, we refer that as a Watson strand, and the bottom strand is referred as a Crick strand. So this basically tells that both Watson and Crick strand, some part is copied as the leading strand and some part is copied as the lagging strand. Lot of students have this misconception that only one strand is copied as a leading and another is copied as a lagging. No, from the origin of replication, it goes bidirectionally, and in one strand the Watson strand is leading and in another the Crick strand is leading. So that is very important concept that students should understand. And the most important aspect of replication is that the replication requires priming. You need primers to do that. There is a similar reaction goes on in the cell, which is transcription, where ribonucleotides are added. There is a polymerization of ribonucleotides, and that is done by RNA polymerase. In replication is a polymerization of deoxyribonucleotide, and that is done by DNA polymerase. So there is no problem in that. But RNA polymerase does not need any primer. Hence, the transcription does not require any priming. But for DNA replication, you need a primer. Why is that? So first, let's talk about the nature of the primer. It turns out that in DNA replication, the nature of the primer is RNA, not DNA. Now, when you do replication in a test tube, like you do PCR, there you make the primers which are DNA primer, and the polymer is able to extend that. So in a test tube, you can have a DNA primer, no problem. But within the cell, it is never that it is a DNA primer, it is always an RNA primer. Why is that? So let's look at it this way. For synthesis of DNA, you need priming. So now, if the nature of the primer is DNA, in order to synthesize that, you need another primer. In order to synthesize that primer, you need another primer, right? So you cannot solve this problem. But if it is RNA primer, then the first primer can be RNA and then it can be synthesized. There is no problem in that, right? So that's why it is not a DNA primer, because by the conditions are such that in order to synthesize a DNA or in order to add any DNA, any DNTPs, you need a priming. Why is that? Why nature developed it in such a stringent way that for DNA replication you need a primer, but for RNA polymerization you do not need. So for that, what we need to understand is that what is priming, exactly what is happening there. So if you look at the sequence here, the sequence written in black font, so suppose that is the template, and then you have the primer which is written in the blue. So the last nucleotide, the 3' most nucleotide, in this case it is C, and which is perfectly hydrogen bonded with the template which is G, right? Now this C has a 3' hydroxyl, and that 3' hydroxyl will act as a primer. And the condition is that the 3' hydroxyl, which is coming from the nucleotide, if that is perfectly hydrogen bonded, then only that will act as a primer. Now look at the next picture where you have G, and in opposite to G there is an A, and it is not properly hydrogen bonded. Now this A has also has 3' hydroxyl, but that 3' hydroxyl cannot act as a primer for the DNA elongation because this A is not properly hydrogen bonded. So that is a boundary condition. Now this way it is making sure that the last nucleotide which is added is the correct one. If the last nucleotide which is added is not the correct one, then further addition of nucleotide will not take place. So this is a proofreading activity coming right there. And if the last DNTP deoxyribonucleotide which is added is not correct one, it is not hydrogen bonded properly, then that will be removed by the 3' to 5' exonuclease activity of the DNA polymerase. Right? So this is the mechanism through which it is making sure that DNA polymerization goes by a high fidelity. There is no error. That's why you need a primer. Now for RNA, such requirements are not there, because even if in RNA some misincorporation takes place, that will not have that kind of deleterious effect. You may tell that, well, RNA is the one from where you get the protein. So if there is a misincorporation in RNA, then there could be a mutation, and in the protein there could be a change in the amino acids, and then activity may go, all those things may happen. Yeah, I do agree with that. But however, these are all temporary phenomena, because RNA has a certain half-life, protein has also a certain half-life, they will be degraded. So the next time the RNA is synthesized, it may not have the error. But if there is an error incorporated into the DNA, then it will not only stay in that organism, it will also be propagated to the progeny, and that change will be permanent, right? So that's why the cell employs many different machineries to make sure that its most important material, which is the genetic material, the DNA, does not have any mutations, right? So now there are two activities going on. One is polymerization and one is exonucleation. Which one wins? The simple thing. The last nucleotide which is added, if it is properly hydrogen bonded, then polymerase activity of DNA pol will win and it will add another nucleotide. If that is not properly hydrogen bonded, then polymerase activity will be blocked. The exonuclease activity of DNA polymerase will win and it will remove that wrongly incorporated nucleotide, right? Okay, so now we'll start on prokaryotic DNA replication. As you know that there are three phases: the initiation, elongation, and termination. Now for the initiation phase, it was discovered that DNA replication takes place it starts at a particular point, and when that region was inspected, it appears that there are certain sequence motifs are there on the DNA. How this was discovered? So basically you take a non-replicating plasmid and you clone different part of E. coli genome into the non-replicating plasmid. And then you see that when this non-replicating plasmid become a replicating plasmid, so it can now replicate. So then you see what part of the E. coli genome was cloned and hence it became a replicating plasmid. So that is the region which is the sequence of origin of replication. So that's how it was discovered. Now it appears that there are certain sequence motifs. There are 5 9-nucleotide motifs, and adjacent to that there is a 13 nucleotide motifs. And in those 5 9-nucleotide motifs, there is a particular protein called DNA A binds. And it is not a single molecule of DNA A bind. It is a barrel of DNA A binds. Lots of DNA A protein binds. And the 13 nucleotide motifs, it is not a motif where protein binds. This is basically 80 rich sequence. So when a barrel of DNA protein binds to this 9 nucleotide motifs, then there is a torsional strain exerted in the neighboring region of DNA. Since this region is highly AT rich, the hydrogen bonds breaks and this region opens up. Right? Once it opens up, then a protein called DNA B, which is a DNA helicase, that now binds to this region and then it melts on both the sides. So DNA helicase is the protein that melts DNA, or from double-stranded DNA it makes to single-stranded DNA, right? But it cannot act on it cannot bind to a fully double-stranded DNA. It needs a single-stranded region where it should bind, and then it will melt the double-stranded region of the DNA, right? So there is another protein is available there, which is called DNAC, which does not have any enzymatic function, but it is kind of, the chauffeur of a car. It brings DNA B, recruits DNA B to that origin of replication. Now, once it is melted, then RNA primer is synthesized by an enzyme called primase, and the length of the RNA primer could be between 4 to 15 nucleotide. And then once this RNA primer is synthesized, then DNA polymerase 3, which is the main replicating enzyme, that will extend this RNA primer and keep copying the DNA template. Now here at this point, one very important thing to know is that when you melt the DNA or when you unwind that DNA, it can readily form hydrogen bonds because you have the complementary bases just in the near vicinity, opposite to each other. It can form the hydrogen bond very rapidly. So these are very sticky in nature. So in order to prevent the hydrogen bond formation, there is a protein called SSB, single-stranded binding protein, that will come and bind to the single-stranded region so that these two single strand cannot come closer to each other and form the hydrogen bond. Okay, so now you look at this very carefully, that you have melted certain region and there is a primer which is synthesized, and this primer is extended, and now the polymerase is here. The polymerase will go only in the 5' to 3' direction. So another primer need to be synthesized here and it will copy this region. Now, if this goes, then you get this kind of thing, and now the polymerase is supposed to be here. It again need to go there and extend this strand, because next step this will be melted, right? So in order to avoid this back and forth movement of polymerases, there is a particular mechanism which is there, which is called trade milling mechanism. Many of you, know, or you go to do trade milling at the gym, or maybe you may have it at your home, where you are running or you are walking several kilometers inside your home without any displacement, because the path is going backward. You are not going forward, you are at the same place, but your path is going backward. The same mechanism happens here also. Once the what are those things? Yeah, so once the leading strand is synthesized, the enzyme is here and it passes the other strand through it so that that strand is copied. So the DNA strand is going backward through the enzyme is stationary there, right? So this way it can synthesize this strand. Now again you have the 3' end, it will be extended, another Okazaki fragment will be formed here, and that's how it goes. Now the question is how to join this Okazaki strand, because DNA is not fragmented after it is replicated. They're all continuous. The leading strand is always continuous and the lagging strand will be continuous after some time. So they will be joined together. Now there is an enzyme called DNA polymerase 1 which comes into play here. So you have the previously synthesized Okazaki fragment, and then this is a newly synthesized DNA. DNA pol 3 is there. When DNA pol 3 sees that there is a RNA primer, it stalls, it does not do any more replication. Then DNA pol 1 comes into the picture. DNA pol 1 is the only polymerase which has a 5' to 3' exonuclease activity. Now, DNA pol 3, it has polymerase activity which is 5' to 3'. It has exonuclease activity which is 3' to 5'. But DNA pol 1, in addition to these two activities, 5' to 3' polymerase and 3' to 5' exonuclease, it also has 5' to 3' exonuclease activity. And through that activity, it removes the RNA primer and maybe some bit of the DNA also. And since it has a 5' to 3' polymerase activity, it will resynthesize that region. So then what will left here is basically one nick. So this phosphorester bond will be sealed by an enzyme called DNA ligase. So that's how DNA is synthesized. Now let's look at a scenario that your DNA replication started at a particular point, and that is called origin of replication. Where it should end? It appears that it also ends at a particular point which is diagonally opposite to the origin of replication. Now other experiments have shown that if you say DNA is not only used as a substrate for DNA replication, as a template for DNA replication, it is also a template for transcription. So what will happen that if both transcription and replications are going together in the same region of the chromosome, then transcription gets the priority. DNA replication stops there. Let the transcription be over, and then again replication begins. So if that is the case, then it is quite possible that suppose there is a transcription going on this side, and this replication fork stalled for some time, and within that time, this left side of the replication fork moved ahead, and they met somewhere here, which is not diagonally opposite but asymmetric place. But it never happens. And the reason is, at this place, which is diagonally opposite to the origin of replication, there are certain sequences are there. Particular DNA sequences are there. That is called terminator sequence, which will terminate the DNA replication fork over there. And in this terminator sequence, there are certain protein called TAS protein binds. And the protein is such that it will allow the replication fork to go through it from one direction, but not from the other direction. So that way, both the replication forks get trapped within this region of 2 TAS protein. So even if one replication fork move ahead, it will go and get stuck there. It cannot go past the TAS protein from the other side. So that's why it is maintained that replication also ends at the same point. Okay, so at this point, it will be very interesting to, clarify some of the concepts, and which you can do by asking your students certain questions. The first question could be that E. coli DNA replicates bidirectionally, and the time required for transit of both replication fork is 40 minutes. Explain then how the mean generation time of E. coli is only 22 minutes. So in 22 minutes, the E. coli divides and the replication takes 40 minutes. So how is it possible? It looks like a puzzle, but in E. coli it is not, because even if one round of replication is over, another round starts, the third round starts, right? So unlike eukaryotes, where you have a licensing factor which monitors that one round of replication is done, then only cell division takes place. Then after that, in the daughter cell, the new replication will start. Here, it does not happen. Here, the replication is continuous. Before one round is completed, the second round starts, the third round starts, and like that. And then when the one round is completed, then you have 2 such molecules. They are now segregated in the daughter cells. But there, as you see that when they're segregated, there is another round, and the next round they have already started. So that way, the mean generation time is 22 minutes, but each round of replication, if you see, then it takes 40 minutes. The second problem could be on the termination thing, that transcription gets priority over the replication and all those things. So this is a map of, is a physical map of E. coli, where all those different genetic loci are marked, A, B, C, D, E, F, G. And from origin, the distance are according to the map. Now, what one expect is that if replication starts from origin, region A will be replicated first, because that is closest to the origin. Then region B, then region C, and the other fork, which is going leftward, it will, after when C is replicated, then G will be replicated from the other side, and from this side D will be replicated, the other side F will be replicated, and finally E will be replicated. So what we expect is the order of replication should be A, B, C, G, D, F, E. In a particular experiment, it was found order is A, then G. Although B is close to the origin, it did not get replicated. So G was replicated, F was replicated, and then B was replicated. I think the reason is very clear to all of you, that which means between A and B, there is transcription going, and hence replication fork stalled over there. The other replication fork continued through G and F, And now if once the transcription is over, then B, C, D, those are, replicated. And between F and E, there is another block. So that means there is another transcription going on between F and E. Hence, after F, it took so long time to replicate region E. And within that time, B, C, D, everything was replicated. So it tells that there are two transcriptional activities was going on, one between A and B and another between F and E. So once you explain this to your students, they will have a very concrete idea about how these things are regulated. Okay, now the third problem, which is also very interesting, we told that for primer synthesis, there is an enzyme called primase, and which synthesizes RNA primer. That means it is a DNA dependent RNA polymerase, right? So in this problem, they're asking us to design an experiment to prove that the primase and not the DNA polymerase 2 makes the primer for precursor fragment during replication. So primase makes the primer, not RNA polymerase 2. So that is the thing you have to prove. Both of them are DNA dependent RNA polymerase. So both of them can use DNA template and it can make RNA, right? But in the cell, why is there is a particular enzyme called primase which is making, not the RNA polymerase which is involved in transcription, right? So the transcriptional RNA polymerase is not involved in replication which makes the RNA primer. How will you prove that? So that the approach should be you inactivate one and then you see that RNA primers are made or not. That is the approach. So you know that there are certain inhibitors of RNA polymerase 2, rifampicin and all. And you can use rifampicin and you see that the transcription is blocked, but DNA replication is not blocked. The primers are synthesized. There's no problem in that. So which means that RNA polymerase 2 is making the primer during replication. Now you do from the other side. You try to knock out the gene. If there is inhibitor available, good. But since there is no inhibitors of primase, you knock out DNA primase, and then you show that replication is not happening. Right? That will be the logic. However, this is not possible because replication is an essential process. If you block replication, cell will die. Right? So you cannot create such mutant. So by definition, you cannot create a mutant of an essential gene. Now, all the genes whose products are involved in DNA replication, polymerase, topoisomerase, or ligase, primase, all those things, you cannot make a knockout because they are essential. Now, what to do about it? There is another way where you can make a conditional knockout. Condition applies, which means you make a mutation and at a particular condition, mostly let's say temperature sensitive mutation. At a particular condition, this mutant does not behave as a mutant, it behaves as a wild type. So you can create the mutant because it will not show any mutant phenotype, it will not die at a permissive temperature. Now, when you have to do the assay, you shift it to the non-permissive temperature. There, the mutant will behave as a mutant. So that way, creating that mutant is not a problem because you can create as a permissive temperature. Let's say at 30 degree, it is a permissive temperature. At 42 degree, it is non-permissive temperature. So there are many such examples out there. So that way, you can create a mutant of DNA RNA primase and conditional mutant and put it in non-permissive temperature and then show that at that temperature there is no RNA primer which is synthesized. So that way you can prove that primase is the enzyme which is involved in making the RNA primer during replication. Okay, so now I have some 25 minutes or so. In that, we'll try to cover certain important aspects of eukaryotic DNA replication. So remember that there are particular sequences at the origin of replication of E. coli. Similarly, people have solved that yeast as a eukaryotic model, that there are also particular sequences at the origin of replication. We call them ARS, autonomously replicating sequences, and there are these sequences called A, B1, B2, B3. A and B1, there is a protein complex called ORC, origin recognition complex. They bind, and there are of such protein ORC1 to ORC6. They bind there. And then this B2 region is the region which is AT rich, and that opens up. Now, for that, you need another protein called ABF1, ARS binding factor 1, which binds to the B3 region, and then upon this binding, this B2 region opens up. So basically the concept is the same like E. coli. There are certain proteins which bind to a sequence and the neighboring region is AT rich and that opens up due to torsional strain. So the concept is the same, but the detailing is different in this case. Now there is I will not go into the detail of this experiment because we don't have that much time. I need to keep some time for discussions also. There is a DNA fiber autoradiogram experiment was performed, and that proved that DNA is in eukaryote also DNA replication is going through the bidirectional mode, not a unidirectional mode. Okay, these are let's skip. Now there are what are the DNA polymerases in eukaryotes? The main DNA polymerase is DNA polymerase delta, which is equivalent to the DNA polymerase 3 of bacteria. Now there is DNA polymerase alpha that is tightly bound to primase and it is involved in priming. So in eukaryote, the primase starts the RNA primer, and then with the primase, DNA poly alpha is also associated. So after the RNA primer is made, then DNA poly alpha makes some bit of extended little bit, and then the main replicating enzyme, which is DNA poly delta, comes into the picture and it starts replicating. Now in eukaryote, there is another DNA polymerase, is called DNA pol gamma, pol G, that is involved in mitochondrial DNA replication. Right. So all these enzymes like DNA pol delta, which is the main replicating enzymes for the nuclear genome, DNA pol gamma, which is the replicating enzyme for mitochondrial genome, all of them have 5' to 3' polymerase activity and 3' to 5' exonuclease activity. However, none of them have 5' to 3' exonuclease activity like DNA pol 1. Now the question is, if that is the case, then which will remove the RNA primer from the DNA, right? So in eukaryote, there are two such models are there. Depending upon which eukaryote you are working on, either of them will find that that is operational. The first one is called flap model. So you have the previously formed Okazaki fragments, and then you have the newly synthesized Okazaki fragments. When DNA pol delta sees that there's a RNA primer, then what it does with the help of a helicase, it push aside. So helicase will melt it, which the DNA double-stranded DNA which is in front of it, and then you have this flap generated, and DNA pol delta keeps on continuing making, copying the DNA, and this flap, which contains RNA primer and bit of the DNA also, will be then removed by an enzyme called FEN1, flap endonuclease 1, FEN1. And then after that, there will be a missing phosphoester bond, which will be sealed by an enzyme called ligase. So the other model is called, it involves another specialized protein called RNase H. RNase H is a protein which will remove RNA from a DNA RNA hybrid. So it does not work on a RNA itself, but is a DNA RNA hybrid, it will remove the RNA part. Now the new Okazaki fragments, when it comes and encounters the RNA primer, then RNase H keeps removing the part of RNA primer. So RNase H can break a phosphoester bond between two ribonucleotides. So it keeps on removing one ribonucleotide at a time. But then the last ribonucleotide, that is in a phosphoester bond with a deoxyribonucleotide, the last one. So that RNase H cannot remove. So then again that enzyme called FEN1 will remove that ribonucleotide, and ligase will seal that. So these are the two mechanisms through which Okazaki fragments are ligated. Now, it will be a good idea to give this kind of summary slide of all the enzymes which are involved. Unfortunately, in both prokaryotes and eukaryotes, the scientists have used those Greek letters for the nomenclature, and they are not the same in prokaryote and eukaryote. So that is a bit confusing. So for example, in prokaryote, DNA pol 3 is the main replicating enzymes, and it has at least 10 subunits. Some of the enzymatic, some of them are the structural subunits and all. Out of that, alpha subunit has the polymerase activity, 5' to 3' polymerase activity. Epsilon subunit has the exonuclease activity, 3' to 5' exonuclease activity. And theta subunit has a structural role. It keeps all the subunits together. Now there are certain accessory factors are also there, like beta. Beta has a side ring clamp activity. So basically, when we will teach polymerase activity, there is another activity which comes into picture, which is called processivity. What is processivity? Processivity is the property of an enzyme through which it can hang on to the DNA. So DNA is a single-stranded DNA, right? So the polymerase, when it's replicate, it should not fall from that DNA, right? If it falls, then there will be truncated DNA synthesis, right? So that's why it hang on to the DNA, it cling on to the DNA with a clamp, which is called beta clamp. So through that, it can, hang to the DNA and it can keep on polymerizing. Now, on the other hand, eukaryotes, their DNA polymerases are not called DNA pol 1, 2, 3. They are called DNA pol delta, which is the main replicating enzymes. DNA pol alpha is also there, but alpha has a limited activity during priming. It is associated with RNA primer, the primase, which makes the RNA primer, and then alpha comes into the picture. And DNA pol gamma, which is the mitochondrial DNA polymerase. And here another important protein is called pCNA, proliferating cellular nuclear antigen, which is the sliding clamp, which performs the function of the beta subunit of DNA pol 3 in E. coli. Okay. So now I'll talk about the last topic of replication, and which is called the end replication problem. So which is very interesting problem actually. Now, there is a concept called Hayflick's limit. Those of you who are doing primary cell culture, you know that there is a Hayflick's limit, which basically tells that primary cells can be subcultured or can be passaged only up to certain generation. After that, they do not grow. And this is something that Hayflick's discovered, and is called Hayflick's limit. So a Russian scientist called Alex Olovnikov, he heard about this Hayflick's limit, who initially worked with Alice Carroll, and then he has his own lab and he discovered that. He also heard Crick talking about the DNA structures and how replication takes place and all. Then one day he was sitting in a metro station and he was observing that, you know, the trains coming and trains going, and he was still sitting there. Then he got an idea that now I know how Hayflick's limit is coming. Why primary cells you cannot culture after certain generations. He came up with this idea that suppose the train, the engine of the train is your polymerase, then if it, and the rails are your DNA, then when it goes, it can copy the entire track, entire railway track, but not the end where it was sitting, because the enzyme is, or the engine is before that. So this region cannot be copied. Then he said that if this happens, then after every replication cycle, after every generation, the chromosome will be shorter and shorter. And then it will hit a particular length, then maybe the essential gene will be lost and cell will not allow that. And that's why after certain generation, after certain passage of primary cells, you cannot, the cells will not replicate, and that is called replicative senescence. So that thing happens. So that was the basic idea he came up with. It's a brilliant idea. It's all, you know, theoretical idea. He thought about those and then he came up with that idea. Exactly similar thing happens in the cell, but the details are a little bit different. So it is not only the RNA primer which is removed and then the chromosome becomes shorter, but there is another aspect is also there. Remember that when we are talking about Okazaki fragment, we talked about that the DNA is unwound in certain installment and DNA replication takes place in that installments, right? In E. coli, the DNA is unwound up to 1 kb 1000 base pair, and at a stage 1000 base pair is copied. Then next 1000 will be melted, that will be copied. It goes like that. But in eukaryote, it is the installment of 200 base pair, and this 200 number is very significant, because if you think that in the histone, the length of DNA that wraps histone is 200, exactly 200 base pair. So basically replication takes place one histone at a time. Okay, so now what happens if at the end of the replication, the length is less than 200? Then another priming event does not take place. So then this region cannot be replicated because another primer cannot be formed here, as this length is less than 200. So in the second generation, you lose this particular length. So that way, you lose lot of sequences at the chromosome ends. This is called end replication problem. How to solve that? So a particular enzyme is there which is called telomerase. The telomerase adds those lost sequences. So at the chromosome end, there is not a particular sequence, there's some repeat sequences are there. We call them telomere repeat or telomere sequence. The telomerase enzyme does that. And now the telomerase enzyme is present in all our, cells, but it is not expressed. It is expressed only in the germline cells, the sperms and the eggs, and also in the stem cells. Otherwise, telomerase enzyme is not expressed. So, since telomerase is not expressed, then with every round of replication, the chromosome length becomes shorter and shorter. So when we are just conceived in our mother's womb, then our telomere length is around 15 kb. At the time of birth, we have already lost 5 kb of our telomere. It is 10 kb. And when we die out of age, so then we have 5 kb telomere, right? But during this time, if somehow telomerase enzyme is become active, then that gives rise to cancer. 85% of the cancers are associated with telomere reactivation. So telomerase enzyme become active, and there is an uncontrolled division cell. Since the chromosome ends are maintained, it is not getting shorter. So there is uncontrolled divisions that takes place. Now there are certain enzymes which are involved in that you may probably tell. That telomerase enzyme, which has its own template, RNA template, which basically adds the telomere lipids and all that you can tell. And your talk should end with the topological problem, which is the topoisomerases. There are two types of topoisomerases which are involved in replication. One is type 1 topoisomerase and another is type 2 topoisomerase. Type 1 topoisomerase, it cuts only one strand. Type 2 topoisomerase cuts both the strand. So typically during elongation phase, when you have to, once you unwind the DNA and you have to relieve that strand, then it is type 1 topoisomerase are used. You cut one strand, pass the other strand through that and delete the strand, and then you again join it. But at the end of the replication, when you have 4 strands together and you need to separate them two strands each, then you need to cut both the strands, and there type 2 topoisomerase comes into picture. Now there are lots of other differences between these two, and somehow this part is a favorite part of those who sets the question paper for multiple choice, and so it will be good to have some kind of table that this shows that what are the type 1 and type 2 topoisomerase, what are the properties and all. Now when I talk about type 1 and type 2, these are the type. Remember that there are many names of topoisomerase. Topoisomerase 1, topoisomerase 3, topoisomerase 5. Those are all type 1 topoisomerase. All the odd numbers are type 1 topoisomerase. Similarly, topoisomerase 2, topoisomerase 4, topoisomerase 6. Those are all type 2 topoisomerase. Even numbers are type 2 topoisomerase. Okay. So I think I will end here. We have some little time. Close to 10 minutes, so we can have some discussion if you have any questions.
Questions and answers for discussion from NRCs across the state. I request all the resource persons and the participants from the different NRCs to interact with the resource person, Professor A. Bhattacharya.
Sir, good morning, sir.
Good morning.
Sir, I am from Srikakulam, sir. NRC. Myself, Common Degree College, Women's Srikakulam.
Okay.
Sir, just I want to know the relation between telomerase and cancer growth. You mentioned, sir, in your talk.
Yes, yes.
I'm not getting that point, sir. Can you
Okay,
please
okay.
clarify it, sir?
Right. So in all our somatic cells, what happened is that the telomerase enzyme is not active. So then after every rounds of replication, the chromosome becomes shorter and shorter. Right. And then once it becomes critically short, then there are checkpoints, retinoblastoma checkpoint, P50C checkpoints. There are many checkpoints which will make sure that the cell division does not take place. There is no replication. So this is called replicative senescence. Okay, maybe if I can show you this graph here. Yeah, is it visible to you now?
Yes, sir. Yes,
Yes.
sir.
Okay. Okay. Let me tell. So in the stem cells and the sperm and eggs, telomerase enzyme is there, and it maintains its telomere length. Now, for most body cells, telomerase is silent, and with cell division, the length decreases, and then it reaches a particular point. Then we call them senescence, replicative senescence. After that, cell division does not take place. Okay. Now, what happens in cancer cells? Telomerase is reactivated, and hence it maintains the telomere length. Okay, since it maintains the telomere length, there is no problem in dividing the cells because telomere length will be maintained. So decrease in telomere length is a signal for stopping the cell division. Now, if telomere length is increased by telomerase, ectopic activity of telomerase, which it's not supposed to do, so then that signal is not there, the cell keep on dividing. That's why you have uncontrolled cell division, which is one of the hallmark of cancer.
Thank you, sir. I got that point. Thank you.
Yeah.
Over to NRC. Have raised their hand.
Sir, this is from NRC Kadapa, women, and your lecture has started with a beautiful question, sir, and it is a very enthusiastic lecture where we can find why we are maintaining 45 minutes after the transformation experiments, while other thing is only 22 minutes of generation, and the differences between eukaryotic and prokaryotic cells, the replication especially, have given elaborately. And till now what we know that only RNA polymerase is going to synthesize the primer, but now we know that there is RNA polymerase 2, which actually play a role in DNA replication in E. coli. And other things.
RNA polymerase 2 does not play any role.
Okay,
RNA
sir.
polymerase 2 is not the enzyme which makes the primer. It is the primase enzyme.
Primase, yes, sir.
Yes, not RNA polymerase 2.
Yes, sir. So we know that no DNA polymerase can initiate the synthesis of the DNA right now. And
Right.
regarding and it is a very excellent lecture, and I do have a doubt to express here, sir. So the question in the beginning of this session is why only 32% of the cells, 32% of is the hybrid DNA, and what is the X you have asked, sir? Probably any inhibitor of these DNA polymerase or helicase, it may be possible that X is, sir.
I think I did not discuss it today.
Yes, sir. You have not
Yeah.
discussed.
Yeah,
but
last time when you guys came, that time I discussed this question
actually,
because today the time was less.
yes, sir.
Right. Last
So,
time I gave talk for 3 days. This is 1 day, so time was little less. So I could not. That is a very important point that you are making, but we could not discuss it today.
okay. What may be the X, sir?
The X is a substance that block initiation.
Okay, sir. Okay, thank you so much, sir.
Yeah, right.
We had a very nice lecture today regarding basic. We feel that it is a very basic topic, but we learned many new things in this lecture, sir. Thank you so much, sir.
Thank you very much.
Sir, you are raised the hand. Sir, generally, no
Actually, I cannot hear properly.
If mobile is not available, what will happen, sir? Then comes to the who proposes the DNA by sir?
I could not hear that. What was the question?
Your voice is not audible, sir. Is not. What will be happened, sir? Your replication.
Sir,
Sorry, I could not.
sir, if topoisomerase is not available, then what will happen in the replication?
See, there are many topoisomerases within the cell. Okay, so if you need to have one or other topoisomerase, otherwise replication will not happen. So there are certain topoisomerases which are absolutely essential. You can only make temperature sensitive mutant, but you cannot knock them out. But there are other topoisomerases that you can knock out, because then some other topoisomerases compensate for their function. But you cannot have any situation where all the topoisomerases are non-functional. Then it will lead to the death of the cell. And hence topoisomerase is a very good target for inhibitors, inhibitor targets for many diseases, bacterial diseases, protozoan parasites and all. For many diseases, topoisomerase is a great target.
Okay, sir. Thank you, sir.
Yeah.
Now, it's time to propose the pharmacopoeia. Thanks to the Professor Nodal and Biochemistry. Over to NRC Kadapa, sir. NRC Kadapa. Please respond.
Sir, responding, sir.
Responding, sir. Sir, proceed, sir.
Sir, this lecture was very enlightened because although it is very basic topic, that is DNA replication and other things, but we learned many things, sir, regarding DNA polymerase 1, 2, 3 in prokaryotes and all other things. And it is more enthusiastic in discussing this telomerase activity also, sir. What is the difference between these cancerous cells and how? We know that cancerous cells grow continuously, that is no stoppage, no senescence, but we know today in this session, we know that why the senescence has not occurred in the cancerous cells. And regarding TAS proteins also, while terminating the DNA replication also, the second problem is very interesting. Why after gene A, then the gene G is replicated. So it is also very interesting, sir, because of this bidirectional replication. And there are two models have been discussed, that is FLAP model and DNA is RNSH model in eukaryotes, where the primer is removed in these cases. And finally, it is a very enthusiastic lecture, and actually we enjoyed a lot in this lecture, sir. Thank you so much, sir, for giving this opportunity.
Thank you very much.
Thank you, sir. Thank you on behalf of all the participants from all the.
Excuse me, sir. Excuse me, sir. Srinivas, sir.
Yes, sir.
Sir, Anil sir came, sir, here. Anil sir want to interact, sir.
Yes, sir. Sir can interact. Sir is there, sir. Still, sir is there.
Sir, very good morning.
Dr. Anil Kumar, sir, from Commissioner of Biology Education is going to interact with you, sir.
Sir, very good morning, Professor Nodal Bhattacharya ji. Really, our faculty are very fortunate to have your lecture. It's a wonderful, and I can appreciate that still you are using the hand-drawn pictorial these things for your presentation. That is really very good. I request all the faculty from Zoology, Biotechnology and Biochemistry These are also available as YouTube links. Please share these links to your students because these are very, very what you call as delivered by high profile professors across University of Hyderabad and some Genome Valley and industry also. So kindly make use of these, all the resources, share with your all students, both botany, zoology, biotechnology, biochemistry, microbiology, so that this can be very useful because highly intellectual people are delivering these talks and these must be available to students as well. Not only that, we will also keep these lectures in our LMS also. So I think all our faculty as well as students must use these highly intellectual resources whenever they feel to listen and they feel to watch this. Really, thank you very much, sir. The University of Hyderabad is supporting us, particularly the life sciences. And I also, on behalf of our commissionerate, I request you to mentor our faculty so that they can also get some projects like DBT and DST also. Thank you very much, sir. Thank you.
Yeah, thank you.
Thank you.
Okay.
Yes, sir. Srinivas sir, we can close this.
Yes, sir. Sir, you can sign off, sir. Sir. Thank you, sir. Thank you so much.
Okay. Okay. Should I log off?
Yeah, yes, sir.
Yeah, yes. Okay, great. Thank you. Have a nice day.
Thank you, sir. Thank you so much. Then, break.
రెండింటిని ఒక దాని తర్వాత ఒకటేనా రెండు కలిపి ఒకటేసారి షేర్ చేయగలరు ఇవి స్క్రీన్ అని కూడా వేసేపే కదా షేర్ స్క్రీన్ షేర్ ఇదే వస్తుంది సార్ రవిశేఖర్ సార్ Nagushan sir, I request the KRPs from all NRCs to respond. Sure, start kaleda. Live lo undi kada mante. Madam sir. వాళ్ళు జాయిన్ అవ్వలేదా
అట్లా మాట్లాడతామంటే
ఎవరు ఫోన్ చేస్తే ఎట్లా ఇంకా స్టార్ట్ అయ్యింది వాళ్ళది ఎక్కడ కనపడలేదు
సెషన్ పెట్టారు కానీ మీరు సెషన్
కరెక్ట్ కొట్టి అది కాదు మన లింక్ కొట్టు లింక్ పార్టీ అయిపోయారు కన్ఫ్యూజన్ లో ఉంటారా లింక్ లేదా ఒకటే లింక్ నాకు వాళ్ళు క్లోజ్ చేసినారు ఏమన్నా లింక్ టచ్ చేయలేదా? వాళ్ళకి వీళ్ళు దీంట్లో షేర్ చేసిన ఇది అంత డెవలప్మెంట్ లేదు ఇంకా. అది నేను
The next session is online mode from NRC Anandapur. The presentation is by Dr. G. Rajshekar sir and writing of scientific papers. So I request all the NRCs and the PRGs be ready. The search person is ready.
Sir, shall I proceed, sir? Sir?
Just a minute, sir.
Sir, is my PPT visible to all?
Sir.
PPT.
Now it is visible, sir.
Yes, thank you so much, sir.
Sir, I will get a clarification from all the answers. Just a minute, sir. NRC, we got one. Are you ready, sir? NRC, Srikakulam. NRC, Vishaka. NRC, Vishaka, please respond once. All the participants are back to their seats of the break, sir. NRC, Tadapur.
Responding, sir.
Okay. So all participants are back, please, sir.
Yes, sir.
Okay.
Sir, this is from NRC Visakhapatnam, sir. We are all present here, sir.
Okay. NRC Guntur. NRC Nellore.
NRC Nellore responding, sir.
Okay, madam.
Sir, please ask the participants to sit in the first rows because I have some questions which are to be answered very fast.
Sure, sir.
Sir, NRC Guntur responding, sir.
Okay. NRC Kadapa, please respond once. NRC Srikakulam also. NRC Srikakulam. And NRC Kadapa, please respond once.
Sir, good morning, sir.
Yes, sir. NRC
Srikakulam.
Okay, sir. So all the participants are ready, sir.
17 members present, one is absent, sir.
Okay, sir. Sir, Rajshekar sir, you can proceed, sir.
Thank you, sir.
So let me introduce you from our side, sir. It is a privilege to introduce one of our colleagues and a highly resourceful person, G. Rajshekar garu from NRC, from GVC Anandapur. He is going to present a talk on the scientific writings. Over to Rajshekar, sir.
Okay, thank you so much, sir. Also, I request from my side the participants to please sit on the first row because this is going to be interactive session. I'm going to share a little knowledge that I have with respect to the scientific writing. Okay, before going to discuss the process of scientific writing, I would like to narrate a story that happened almost 8 years ago. I'm going to connect this to the scientific research. Okay. First of all, let me start with an event that happened very long back, almost 1929. A rare event, a rare chance happened in the field of medicine, which had an immense effect after the discovery. You all know that Alexander Fleming, and what is the story? So he worked in a laboratory in the London. So when he was working in the laboratory, he was working on Staphylococcus bacteria. And during his work, he took some leave and he went to for a treatment. He came back to his laboratory and he observed the Petri dishes which contains the bacteria. So on Petri dishes, he inoculated some bacteria and he observed the growth of the bacteria on those plates. So very surprisingly, in one plate, he observed growing up mycelia. He observed growing up fungus. Also, he found that where the fungus was grown in that plate, he did not find bacterial colonies. So he assumed that maybe the extract of the penicillin might have restricted the growth of the bacteria. So he tried to isolate the compound from this fungus, but he could not succeed in isolation of this penicillin from this penicillium fungus. So he, after finding the mycelium on the plate, so he went on to find out what was that particular fungus, and he found that there's penicillium rostrum. So he found that that was a penicillium rostrum. So it happened in the 1929. Okay, so it's completely a rare or a chance event, I could say. So what I mean to say, in the science, majority discoveries are not planned. They are completely unplanned, and unplanned discoveries are more in the science. Even that PCR, so you know that Carl Mullis, who invented the PCR, but his intention was not to develop a PCR for amplifying the DNA, but his intention was something different, and unexpectedly, accidentally, he found that bacteria has one bacteria, an enzyme which is collected from a bacteria has the potential to even function at high temperatures. So that finally led to the developing the PCR machine. You all know that. So coming to these three persons were also very important. These three persons, they also worked on the same penicillium. But when Alexander Fleming, he failed to isolate the compound. So this forward chlorine, this chlorine, Heatley and Chain, who were the professors in the Oxford University in the London, and Fleming, he worked completely in a different laboratory. He was not in the Oxford University. So Florey, Heatley, and Chain, they worked on isolation of this penicillium from the penicillin. And when the Florey asked his colleagues, that Heatley and Chain, to grow this fungus in the laboratory, so the penicillium rostrum they cultivated in their laboratory, but they did not show much growth in the penicillium rostrum. So rather than this one, they found another strain. So the penicillin, which is used for synthesis of penicillium, is not the one which is identified by the Fleming. So a different strain, that is nostrum. So Penicillium notatum, sorry. So that was grown by these three persons, and they successfully in growing it, and also the chain actually he has done it very fantastic work in isolating this compound from the this fungus. Okay, almost it took 10 years for them to isolate this compound from the this penicillium. So 1939, finally they could able to synthesize this compound, the isolated compound. Then in 1945, they submitted this paper to Lancet, which is that very reputed journal. They submitted for Lancet, and it was published in the Lancet, and also received Nobel Prize along with Alexander Fleming in the 1945 itself. Okay. So this fluorine heat leach chain, they have not only isolated the compound, also they injected this penicillium, which was extracted from their fungal site to the mice. So they took the two groups of mice, control and the treated. So both mice were infected with streptococcus bacteria. So they injected this penicillium extract to the treated treatment group. So only the treatment group survived after the experiment. Only the treated group survived, and it was a miracle at the time because there were World War II was going on one side. There was a huge demand, huge pressure on the scientists to find out antibacterial compounds. Okay, now let me go to the session that I am going to talk for next 1 hour. Actually, I have a lot. I want to explain the whole process of writing the scientific paper. I don't think so that this time is enough for explaining all those things. I will try my best to explain the whole thing. If it's not possible in this session, definitely in some other session, I will find a time to complete the remaining part. Just remind me if I exceed the time. Okay. So now I have a question here for all the teachers who are participating in this 5-day training program. So what do teachers need for inculcating a scientific or research temper among students? You please tell me what you want and what the government is to provide for colleges or has to provide the teachers so that the teachers can inculcate the scientific temper. I need answers for this. What are you expecting from the government or from anyone so that you can inculcate the scientific temper among the students? Srinivas sir, are you there?
Minimum equipment, sir.
Okay. What else? Minimum equipment. Only equipment. Are there any other things that are required by teachers to promote scientific temper in students? Okay. See, even we don't need sometimes. So basically, yeah, we need the equipment and all. I agree with you, madam. But because of not having all these, we cannot get an excuse from this one. So there are ways to motivate the students, or there are ways to motivate ourselves to be in this field of the scientific research. For example, when I was in a degree, I think it was in my second year, my teacher is also attending this workshop here. His name Lakshmati sir. He conducted an experiment. Actually, it was not in part of the syllabus at that time. It was not part of the syllabus also. When he took some colored solutions and there was a calorimeter in the laboratory, and he performed one simple experiment, and he asked all the students of my batch to find out the concentration of unknown samples by giving the known samples concentration. So that actually fascinated me at the time, because a simple experiment just with a calorimetry and a colored solution, we could really seek the attention of the students towards the science. And not just that one, even a simple algebraic equation, all that linear formula or linear equation in the algebra, that small equation is enough. I feel that really, if we have intention, or if we are willing and we are aiming to improve the scientific temper in the students. And I have some more questions for you. First of all, I want to say what is the misconception that most of the teachers or most of the researchers have. So the misconception is that writing and publishing in reputed scientific journals needs only good English writing skills. And majority of the teachers or majority of the scientists, they assume that having good English writing skills is enough to publish their article in a reputed journals. This is a misconception. And what is the truth there? Let us see what is the truth there also. The research or academic publications of authors from the English mother tongue countries are also rejected by an editorial board, citing the language needs improvement. That means even the scientists or the researchers who are in the English speaking countries, so though they have a lot of command over the language, still their publications are rejected by the editorial committee. So it emphasizes that, so just having enough English writing skills are not enough. So having English writing skills are not enough for publishing in the journal. That means there is a systematic way of writing. It is not like just writing something that English, we can write paragraphs and can submit, and so that the reviewers will accept our paper. It's not like that. So we should follow a systematic process while writing the scientific scientific articles. So that we are going to discuss in next few minutes. So what is the positive thing that we can take from this truth? So the positive thing that I take from this truth, in my case, I feel that, okay, they also have the same problem that I have. So what my problem is, my journals or my articles are also being rejected. They are being rejected by the scientific community. That means their journals are also being rejected and my journals are also being rejected. So I have this problem that they have. So that means we both have similar problem. So that is the positive thing we can feel from this. What is the negative thing? It's a waste of even trying to understand and learn it. See, when you see the other side of this one, see even people who live in the English speaking countries, they cannot write a scientific paper. How I am from a country where the English is not a native language, how I can try and write a research article. So we should not see this side, we should see only the other side. That means they also have the similar problem that what we are facing. So these are the some points that you ask yourself. So please, I request all the participants to read these points that are present on the slide. See, the first one, is it necessary or mandatory for you to submit a research proposal or research article? Is it necessary to all the teachers who are participating in this 5 days training program? Is it necessary for you writing a research proposal or research article? Participants from other NRCs, can you hear me? My question is it necessary or mandatory for you to write a research proposal or a research article and publish in a journal. Okay. Sir, Srinivas sir, are you there? Sir, Srinivas sir, are you following me?
Yes, sir, I am there, sir.
Yes, sir. I request the participants to answer this one. Without answering for these questions, I can't move for further discussion because these are very important. If at all they feel it is not necessary, there is no meaning of discussing and going the things about the research or scientific writing. If they feel necessary, I need an answer why it is necessary for you.
Sir, Dr. Rashekar Garu, this is Dr. Y. Polinaidu from NRC Srikakulam, sir.
Hi, sir. Yeah.
Sir, as you said, is it necessary or mandatory? It is necessary, sir. In addition to that, as per APSI, the burden of syllabus for on teachers or even on students is nowadays very less time and a heavy syllabus is there, sir. In addition to that, to do research, we need to spend lot of time, sir. So research is important, but we have to think about the time management also. To inculcate scientific temper, we must accommodate some hours for this research, sir. Not only in internship or so, what you call CSP projects. Research is completely different from those projects, sir.
Okay, sir.
So I'm sure that I agree with you. Research is important. And last question: how many of you are agree with that scientific writing is to be included in UG syllabus to make students understand the importance of course or improve their knowledge, sir?
Yes, sir.
That is important. Yes, sir. You have to include, but we have to at the same time we have to give some time for teachers and students to do research in that regular period. In Sundays or summer, do research. It is not like that, sir. Research is a continuous process. We have to give some time to students and teachers in addition to this academic exams, mid exams, semester end exams, evaluation. We are with so many works, sir. So many works are there in our hand, sir. In addition to that, if you want to write a paper within 2 to 3 days, we can write a paper, sir, without doing any research. Nowadays, so many papers are like that, only scientific papers. Not going to comment anybody here, but so many papers are like that only. So one person's profile, if you observe, so many papers are there. But if you observe the timing, how he can able to do all these things within a single year? If 10 publications are there, is it real, sir? Real publications, sir?
Okay, sir. Those are not relevant to this one. Okay. Anyway, I'm going to address those aspects in the end of this session. I'm going to discuss how the UGC, if you see on one side, UGC also changing its regulations year on year and asking and mandating the quality output. So definitely we'll see certain changes in the coming years that we'll discuss later. So let's talk about only the positive aspects of these things, not the negative aspects. Okay. So as Sir said, yes, it is necessary. He is saying that it is very necessary process, but yeah, it is necessary because you have to publish the papers and you have to get the CAS that everyone we agree openly, nothing to hide. Okay, so how many of you agree that scientific writing is to be included in syllabus? Is said yes, it is required. And how many of you felt that you should have been taught this in your academic studies at any stage, like you UG or PG or you PhD? So this is a major problem, and everyone talks about the importance of scientific writing and the difficulties that arise while writing the research paper. But no one comes forward to frame the syllabus and to teach this one. So I don't know the reason for it. Okay. Then how many of you want someone else to write research papers or books in your name? How many of you want someone else to write research papers or books in your name? It seems everyone wants someone else to work for us. Okay, because we have a lot of things to do in our college. So if someone else write for us and keep our name. So we all feel very happy for it. Yes, there are ways, but I'm not saying the first. We can approach the some other persons to help us in this matter. I will show some artificial intelligence tools, AI tools, which you can use. So those tools will minimize your burden and will make the process more easier. So probably I will explain few tools which you can use for writing. Okay. Next, how many of you write daily at least a paragraph having 200 words? Anything that could be any academic academic material, or it could be research material. How many of you are willing to write at least a paragraph having 200 words daily? You ask yourself, and how many hours do you write in a month? So of course you must write at least 1 or 2 hours per week, because writing improves your understanding and presentation of your content. Why I am stressing that? Why I am stressing the point of writing again and again? See, this should be included. Why it should be included at the UG level? Why I am stressing that? Because when the student writes on his own, so his understanding of that subject will be more. See, that's why even when we practice it, and definitely we see the change in understanding of the subject. And kindly don't use the AI tools for writing. Completely don't depend on the AI tools for writing. Just use them for improvising your content. And majority of the teachers, unfortunately, teachers who can write on themselves, on their own, teachers who have very good knowledge in writing, they're also now start using these AI tools for writing the articles. It's really not good for the academic community. Just use them for improvising your content, or getting some ideas. So you can use it. There is no problem in that matter. So coming to my topic, here I'm not to say what why it should be included, whether it is important or not. So those aspects are not important for me. I just want to focus few things which makes you to write on your own. So for example, from this lecture you will understand the structure of scientific research articles, including the different elements of the article and their importance with simple and suitable examples. I'm not going to use the complex examples here. I will use very basic examples from zoology and basic examples from microbiology to explain how to use this process. And strategies for overcoming initial obstacles in scientific writing. It is very common for a beginner. So it's very common. We face a lot of problems in the construction of a sentence, or to have a flow in writing and to connect one idea with the other idea. So we face a lot of problems. So definitely these things will be gradually decrease once you start practicing. So then overview of AI tools and open source softwares that are useful for writing your scientific articles. Then techniques for reading and understanding research papers without reading them in their entirely. So whenever we see a research paper having some 20 pages and 30 papers, so immediately we don't read the article, and we skip that article, and we'll find out article which has less papers, and so that we can read that article in a less time. So that's not correct. So even articles which are some 20 or 30 pages can be read in a very short time if we follow a particular strategy or a technique. These are the takeaways of this lecture. I think if time permits, I will cover all those aspects. If not possible, definitely in the next session, in the coming 2 days, I will definitely try to finish this one. And these are the two things which are being stressed by our CC officials in very recently, research proposal submission and research article submission. Let us see what are the similarities between the research proposals and research articles. The last 2 days we have been discussing here the genome analysis, the sequencing and analysis. There we discussed a few bioinformatics tools to find out the similarity between two different between two sequences, two DNA sequences. So the same way you can see research proposal and research articles and see the similarity you find. There is a lot of similarity is existing between the research proposal and the research article. For example, if you take a research proposal, it starts with the project summary. So what project summary signifies or what project summary contains? So it contains the brief information of the whole proposal. Contains the brief information. So main points containing the main points. So while the research article, it starts with the abstract. There you find again the main points of your complete work. So coming to the research proposal, the second aspect of the research proposal is the objectives. So in case of the research article is the introduction. So in research article, you find objectives in the section of the introduction itself. So in the introduction, we mention the objectives of our study. Okay, coming to the research proposal, there the third aspect is the output of and outcomes of your project. These two are very important. So this section is very important in research proposal to be awarded the grant by the granting agency, output and outcomes. So in output, they expect what are the results in the form of both research papers, seminars and workshops and all. In the aspect of outcomes, they look into the what is the impact that has in that field. So that is very important that you have to find a topic which has a impact in that particular field. So many lecturers who submit their proposals for granting agencies, they allege that no granting agencies are not generous in giving research projects to the faculty members. So the other side, if you see the research article there, the introduction section itself contains the significance of the study, which is very important. The significance of study again explains the impact of your research work in that particular field. Next, research proposal. Under the research proposal, the state of the art. So the state of the art means you have to briefly explain what is the current knowledge in your research area. So in the state of the art, if you start with some with old literature of 3 decades ago or 4 decades ago, so that will not be anyway helpful for your project to be selected by the granting agency. So the reviewer, they only look into the recent findings in your area of research. So in the research article, there also we write the state of the art. So there are that means the literature. So that is a part of the introduction again. So coming to the research proposal, the next thing is the origin of the proposal. It is a very important point, an important section, origin of the proposal. So how you have got idea of something that you want to work in your laboratory. So the origin of the, that means when you look into the literature, you find something and you may not convince with them, or sometimes you may feel that the study is incompleted and need to be carried some more work in that field. So such things you find when you look into the literature. So those things you have to highlight in your research proposals, the origin of the proposal section. There you have to write all these things. Under research article, in the introduction, there we write research gap and hypothesis, so which is similar to origin of the proposal only. And next in research proposal, we write the work plan, which contains the methodology and work time schedule. Also, there you have to provide for granting agency. If you provide your time schedule like 10 years or 15 years, citing that you have so many administrative work and all, so they don't accept it. So maximum the time period for the major projects at any case is 3 years. For minor projects, it is 1 year only. So next under research article, we also write the methodology. So here also we have to write the methodology in research article. So both have the same thing. So up to here we can find some major similarity between these two. And next sections, if you see there, we find the difference between these two things. So expertise. So when you submit a proposal, they ask you to write the expertise that you have in this field. Suppose you have published a research article in the field of the toxicology, and if you propose a plan which works on the genomics of some organisms, so definitely they don't consider, because though your idea is very good, you have a very good proposal, you have identified a very good point with respect to the origin of the proposal, and you have very good outcome. So in spite of all these, so they reject your proposal, citing that principal investigator doesn't have much experience in this field, so they don't. I mean to say, they don't invest on this kind of thing, because anyway, you know that the research grant that we get are completely are the tax-based money. They don't want to do some trials or the experiments with that very precious amount available for helping the researchers. The second one is the publications. They ask us to provide the publications that we have published in the past few years. If you provide last 5 years, that's enough. Sometimes our principal investigators, they assume that if they see more number of articles under publication section, so that our proposal will be accepted by the granting agency. It is completely myth. So they just look into the last 5 years publications, whether the principal investigator has done something in this field of area or not. They just look into for that and see. Also, they look into whether they published in a very good reputed journals or not. Okay. So the other aspects they inquire from the principal investigator are instruments available with the PI and in his neighborhood. So because if they feel that everything is okay with the objectives and state of the art output and outcome, and they decide to grant money, so they look into the requirements of the PI, what he requires. So sometimes you know that our research investigators or the faculty who apply for these grants, they ask a list having all the things, and sometimes they may not be even required for their research study. Also, they write in the proposal. So they look into this one, and if the same equipment is available with the PI institution or a nearby institution, so they don't consider that is required by the principal investigator to carry the work. So whatever the amount you propose to conduct this, okay, they reduce this amount and they sanction the project. So they also ask the budget details to know how much you required and all the things. So budget details also we provide in the research proposal. So it comes to the research article. After methodology, we have a completely different story. There we write the result after doing the experiments, after conducting the experiments. So we get results. So those results are to be written in the results section. Next, we have to discuss the results. Okay. Anyway, I'm going to explain each and every aspect of this research article in detail in the next few minutes. Okay. Finally, conclusion section. So these are the things you can see. Find so many of things are very similar between the research proposal and research article. Very few things are different from each other. Okay, let's move to the next slide. So, introduction. So, I just want to know your understanding about the introduction section of research article. See, for example, there are 3 points or questions that I am asking all the participants to read and answer it. So what should be written in this section? What should be written in the introduction section? Why should they be written in this introduction section? If you know the answer to the above two, what is the order to be followed while writing this section? Suppose you know what should be written, So you should know also which one should be written first and which one should be written the second and like that. And also while you confirm that these are things are required for writing introduction session, you should also have a reason for writing them in the introduction session. So I just want to know your response regarding this so that I could understand what you know already about these points, and I'll try to explain the things that you don't know in this regarding writing the introduction session so that our time, we can manage time very effectively. I can cover most of the so many things. Otherwise, if I explain each and everything, if you know already them, so it is the waste of time for us. Sir, Srinivas sir, I want at least one participant from each NRC to answer this.
So I request all the participants from different NRCs, please respond.
Sir, as you mentioned in your previous line, sir, what is the present status of our study and
Yes,
what
sir.
are the gaps and review of literature, like that only we are going to give some introduction about our topic. Sir.
Okay, you said we should write literature review under introduction section. Why should write?
Regarding study area and our topic and any research gap like that we are going.
Okay, okay. From other NRC?
I request all the participants from the NRCs with the response.
Hello, sir. Good morning.
Sir, good morning, sir.
Sir, NRC VS Krishna Government Degree College for Women.
Yes, sir.
Sir, in your earlier slide, that
Okay,
difference between that writing of the articles, yes, the difference different elements of research paper and research proposal.
Yes.
But here you can mention about that research article. Sir, what is the difference between that research paper and research article? Both are same or is any difference?
Both are same, sir. You can use them any. Wait. Okay. Sorry, sir. There is a mistake. So you can use.
No, sir. No, not mistake. Actually, we are confusing about this article and paper. Research article and research paper. Is there any difference between these two?
Sir, for this question, I will answer at the end. My first things that you have to answer these. I will answer that one. That is not
Okay, sir. Okay.
Introduction. I just want to know what should be written in this section and why should they be written in this section. If you know the answer, please tell them, and also the sequence that we have to follow while writing all the elements in the introduction section.
Sir, is it
Requests from other NRCs also.
Sir, Rajshekar sir.
Yes, madam.
In introduction, we have to give about some significance. Why we are going to do this? What is the importance of that particular study?
Significance, madam. Okay. When should we write, madam? In significance, we should start with the significance.
So for example, if we are writing a proposal, why I selected this topic?
Okay.
That I have to introduce, I have to keep in the introductions.
Yes.
Is it right, sir?
Madam, I will clarify your doubt. So, okay, significance. You said that we have to write significance. We have to start with the significance you are saying, and the reason you are saying that it explains the reason for conducting that particular research. Am I right, madam?
Yes.
Yeah. So other NRC. Okay. Coming to this introduction section, what should we write? Let's know now. Let us understand that. Huh?
Don't expect interaction. Let's go ahead with the
జయ ఇక్కడ రావు దీన్ని ఇంకొక తీయాలంటే ఓకే ఓకే ఉంది. Okay, before going to explain the different elements, okay, just want all the members to see this image and write your observation. After seeing this image, what you have observed, please write down. Okay, shall I move to next slide? Or you need some more time for observing this image? Okay. Shall I move to next slide? Okay. Okay. Please keep this in your mind. I'm not going to say anything about this now. So after 5 minutes, we'll come back to here and we'll discuss this. And next, let me explain. Okay. Sir, Srinivas sir, is Excel sheet visible to all of you? Is Excel sheet from my screen?
Yes, sir.
Part one is visible, sir. Section, element, purpose.
Okay, thank you so much, sir.
So those are visible. Column
Yeah.
A, B, C, D, and E are visible, sir.
Okay, thank you. So there may be few participants who may have more knowledge about this. So I'm just explaining the basic things. Don't mind. So also excuse me if I make some mistakes while explaining this, because so many things are there to explain. So without my consciousness, I may make some mistakes here and there. Please rectify me wherever you find it. Okay. I'm going to start with the introduction. Actually, when you see a research paper, so there you find abstract, but I'm not starting with the abstract. There is a reason for not explaining the abstract at first. So I will explain that later. Okay, now I'm going to discuss about this introduction section, what it should have. So this is the research paper blueprint I prepared for writing the research article or scientific paper. Okay. So what is the first element that is required that is to be in your introduction section is the importance of the topic. That should be there in your introduction part. So it must start with the importance of the topic. So we also call this as a hook statement for this introduction section paragraph, first paragraph. Introduction section's first paragraph, hook statement, we call this also. Okay. How many types of hook statements we can use for writing this introduction section? There may be so many, but we can use a few things which are related to our subject of study. Some people may work in the ecology, some may work in the evolution, some are on ecology evolution. So different things we work on. So few hook statements here I am going to explain to you to understand or to explain the importance of our work to the reader. Why are we writing the research paper? First of all, we are writing the research paper to explain or to explain to the others. See, because we have conducted research paper, we know each and everything, even if there is no book statement, if there is no research gap. So it doesn't make any difference for us in understanding the research problem that we are studying. But when you publish your article, there's some other person who is not directly involved in your work, who is away from you, and he has to understand that information or the results of findings of your research study. So definitely it is a very challenging for us to write a research article. Okay. The first statement is must explain the importance of your research topic. must explain the importance of the research topic. So here we see some hook statements for this one. For example, we can give some surprising facts or the statistics about your research topic. So as Madam answered that that should explain the significance. See, yeah, it's the significance, but it's not completely the significance of the research problem, but the research area. So we find two significances in a introduction section. One significance is to explain the importance of the research area. The other one is to explain the problem, the research problem in that particular research area. See, first one is we start with the broader significance. Okay, so surprising fact or statistics. For example, in India, if you find in the last 2 years, the deaths of the cancer patients has increased. You find that in the last 2 years, the deaths of the cancer patients is increased. So you say that yes, you can by saying this, first 2 lines about the statistics or the surprising facts about your work, you will grab the attention of the reader. So reader will understand that there is a problem, so which is to be addressed or which is to be studied. Okay, that is the one. The other one is making a strong opinion. For example, you can see the example also. Now I'm making a opinion statement. For example, see this one. Chemical drugs used for therapy of diabetes have a strong adverse effects on the patients, causing more ailments. So what is your opinion here? There is no statistics available for this one, that chemical drugs have some adverse effects while treating the diabetic patient. There is no data you have, no information you have. But you have opinion that the drugs have some adverse effects on the individuals and causing some problems, adverse effects to them. So you can write this statement, your first statement like this, so that the readers will attract and they will start reading your paper. The other one is mentioning the problem. See, the first one is also problem, but we are explaining the severity of the problem there. Severity, what the severity is there. The third condition is different. There you see, with the increasing use of antibiotics, antibiotic resistant organisms are formed, making the microbes resistant to the currently available antibiotics. The problem is here, since we have been using the antibiotics for treating of these for treating the bacterial infections. So these antibiotics are also not only curing the infections, are also making the pathogens resistant towards these antibacterial agents. So here we are saying that there is a problem with respect to these antibiotics usage in the society. We are mentioning here. The fourth one is completely imaginary hook statement. It is imaginary hook statement. See, let's see what is the imaginary statement that I have made here. The current COVID-19 virus infects individuals through the air, and it has been found that the infection has a more impact on the respiratory system. Imagine if the new mutations make organisms infect the nervous system, the severity of the outbreak due to the COVID-19 is unpredictable. So there are no evidences for substantiating this COVID-19 infection or COVID-19 severity in the individuals with respect to the nervous system right now. But the author is assuming that maybe in future mutations make the COVID-19 to even infect to the brain tissue and make some changes there, and that will have a very deleterious effect in coming days. So this imaginary statement. So why we have to write all these kind of sentences in the beginning, just to attract the attention of the reader so that he will not skip away immediately after reading the 2 or 3 lines of our article. Why they should read our article? If they read our article, then only our article will be cited, or they will take our article for further reference, so that the citation of our article will increase and the citation of that particular journal also will increase. Got it? So that finally will have impact more on the impact factor of that particular journal also. Okay. So as you know that in movies, the beginning scenes are the introductory scenes which are very important for the success of the movie. So in research paper also, these hook statements, they play similar role. Similar role. Okay. So what is to be written after this? After writing the hook statement, which provides the broader significance of your topic of research area, the next one is, I'm getting some problem with the using of this Excel sheet here. Okay. The next component that we have to write is the literature of the studies of studies that have focused on our area of research. Why should we discuss the literature part here? What is the need of discussing it here? Why should we discuss the literature where we have our own idea? We have very good research problem to be tested or to be verified. Then why should we look into the works of the other persons? Is it to get or steal the ideas from their previous articles, or to copy paste the information from those articles? I need answer for this one also.
Sir, to know the current status of our research topic, sir.
Very good, sir. Very good.
A contemporary knowledge must be needed for a researcher, sir. So for that only we are going to hold on.
Very good, sir. Thank you. See, we have to be very careful while writing this section. So in some research papers, when we read with few research papers, we cannot understand after reading immediately. Sometimes we will need to read twice or thrice also for understanding what is there and what exactly about that paper. So why it happens with only few papers? Because the way they follow, or the way they have they followed writing is probably not not in line with the scientific writing. Even sometimes those papers which are not quality and which don't follow a proper guidelines may be accepted by the very good reviewed journals also. So that happens sometimes. As I said in the first time, that is when a rare event happened in the medicine, like Fleming's experiment. So these are also some rare events. Don't expect that all the articles with that level of quality will get be published. Okay. So while writing this part of the literature, you have to start with a general information regarding your research area. For example, you are talking, you are going to work on some DNA. Yes, you are going to work on enzyme that involved in the DNA replication. So definitely you should write that DNA is a double-stranded molecule in your literature. You should start writing with the DNA is a double-stranded molecule. And some people may argue that that is very well-known thing. Everyone knows that DNA is a double-stranded, and why should we write? So it's not like that. See, sometimes the basic information for researchers who are new to the research field and who start reading the research papers. So for them, definitely this basic information helps them a lot and makes them to understand what actually the study is about. And also it is a kind of respect that we give to the earlier people who have done the work. Okay. And we have to go from the general to the specific. And in the first paragraph, as I said, you have to start the book statement followed by the literature of that particular area. Then we have to, in the literature itself, we have to write the general information. Then you have to go to the specific information. I will also show the example of a paragraph which has this general information to specific transition. Okay. So also at the end of this paragraph, we have to point out the limitations in the previous study. That is very important. Not why I'm saying we should go to the specific, from general to specific. So as I said, only the previous 5 publications are only considered while reviewing your research proposal due to the because the previous 5 articles explains your expertise and how you are connected to that particular area and all. The same way, so when you look into the specific information of that research area, so there only will find the information of what is lacking in the previous studies and what is not properly done in the previous studies. So that information we can retrieve from the only from the section that is the like limitations, or the sometimes you see in some research articles in the conclusion or they write future suggestions. Future suggestions, we see that. So future suggestions means that the researchers who are interested in this, they can work on these lines so that the information with respect to that aspect will be known to the scientific community. Okay. Let me show you how to go from general to the specific. Okay, at the end of the first and second paragraphs, the limitations are mentioned after specific details of the topic sentence. See, sometimes you may not find limitations immediately after the literature. So they will start again a new paragraph explaining some different aspect. At least you may find it that limitation at the end of the second paragraph. Okay. Let's see how we should write a paragraph having general information to the specific information. First of all, see the general information, like you can write digestion to reproduction and immune system. If you want to study some plant extract with respect to these things, so you can write, see with how it affects the digestion and how it has the effect on reproduction, immune system, behavioral aspects of organism, growth aspects of organism, genomic and proteomic and disease progression, etc. So these are all you looked into the previous literature. You have after looking into previous literature, you have found information that yes, this extract has some effect with respect to the physiological systems or behavioral aspects, growth, genomic and proteomics aspects. So this information we write at the beginning. Okay. So finally, when it comes into the genomics, there we talk about much about the genomics. So you write more experiments or the research work that have done in this specific genomics area with respect to that particular plant extract. Got it? See, initially we write all the aspects. It's not required to write all that, but when you write, so the reader will understand, okay, these many areas where this particular plant extract has been studied, so he will get that idea. So after that, you can specify a particular area by taking either genomics, or you can take anything like a digestion or proteomics. You can take any one aspect and then you start elaborating that particular area so that you can narrow down from general to specific, that one. Okay. While writing this introduction session, so you must know what are the tenses, English tenses that are to be used. For example, when you cite only one particular, if you cite one particular study, you can use the past tense for it. For example, Smith investigated the causes of colon cancer. and you are trying to say few generalized things, you can use the simple present tense. And you want to write the experiments of 2 or 3 persons, so then you have to use the present perfect tense for it. Okay, so in some articles they don't follow these no tenses properly, and they write sometimes even for a single study they write in past tense, for even multiple studies which reports that particular issue they write past tense like that. So you find it like that, but don't assume that we can also write the same way and the paper will get be accepted. So that will not help you to actually understand the proper scientific writing. Okay. Next, what is the time now, sir? This is the more important one. Time is 12:55. Okay, I think, Sir Srinivasan, sir, we have another 20 minutes time. Am I right, sir?
Sir.
Yes, thank you so much. Okay. This is a very important area, research gap. Finding the research gap. So actually it provides justification for your research work, why you want to work on that particular aspect is justified by the research gap. Okay. So this research gap contains so many 3 sections actually. Before going to find the sections of this research gap, let me show you how to find it, the research gap that is very required. See, if I explain what are the elements are there, you may ask again how to find it, that one. So I know that is very important. Okay, sir, is my PPT visible now? I'm switching from Excel to
Yes, sir.
PPT
Visible,
now.
sir. Visible,
Yeah.
sir.
Thank you so much. Now I want to come back to this section where I have shown this giraffe. Yeah. Let me tell, please answer me what you observed from this image. What
There is
are you observed from this image?
Sir, survival of the fittest is the basic concept behind this, sir. To get that food, elongation of neck is observed in due course of time.
Okay, sir. Very good. Think of what else? Not in the aspects of the academics, just look into the aspects of the research and answer.
So this research point of view, so the diagram shows the increasing of the knowledge levels.
Okay.
Levels to learning level of the articles, paper submissions.
Okay. Okay. I don't want to say that what you were answered is incorrect. Okay. Fine. What you answered is correct. So coming to this image, okay. When you want to find out a research problem, you have to look into the research literature, because as I said, when you look into the research literature, they will find some limitations in the previous studies, or some future suggestions for your research work, or sometimes even you may not agree with the findings of some people, as Sir pointed out that some researchers, without conducting the experiments, they publish the articles in the journals. In such case, you can perform experiment and disprove them also. That is also there in the research. You can disprove others also with your research work, rather than commenting and making it a issue. Okay. So coming to this one, so when you see here, you find 2 kinds of giraffes. I think so. One with the short neck and one with the long neck. Am I right? So when you see the literature, if the literature is completely explaining about only the giraffes of long necks with respect to their behavior, growth, or some ecological things or evolutionary aspects. So why the previous research has not focused on the small giraffe? See, when you look into the literature review, you got only the results that have focused on the long neck giraffes, not on the short neck giraffes. Why did not they do any study with respect to this one? So that is the problem. That is the gap that you find it from the research, from your previous researchers. That means all the previous studies, they focused on only the long neck giraffes, but not on the short neck giraffes, which are also important, or we can see which are also there. Am I right, sir? So that becomes the gap. That becomes the gap. Got it? So to find out a gap, so what is first required? We have to look into the literature and see what all they have done in this particular thing and note down what all they have not done in that particular aspect. Okay. So now we have research gap. So this convinces your research proposal peer committee or the editorial committee of the research publication. Yes, this is the area which was not focused and needs to be studied. When you convince them that yes, this is a study to be focused or to be studied, definitely the research granting agency or the reviewer of a research journal, they will accept your publication or your proposal. Got it? So what is more important research problem? We don't see or we don't look into the literature. Sometimes we read one or two articles and come to a conclusion, yes, these people, they have not done this thing, and we start doing it and we start we find this as a gap and we write a proposal and submit to granting agency, and they look into your research gap and they immediately reject your proposal. That they understand, okay, the investigator, he did not look into the completely the previous researcher's findings. If it is article, they will simply reject this article is not suitable for this journal. Got it, sir? So the research gap signifies or it explains the justification for your work. Got it? Anyone have any? Does anyone have doubt with respect to these things?
Excuse me, sir.
Yes, sir.
Sir, NRC Visakhapatnam.
Yes, sir. Please, sir.
Sir, that earlier slide, that Shakira et al. That et al. It must be italic or
Sir, those
word.
things, those are small things. I will definitely answer those things at the end. And you need not to worry about these things. Now we have AI tools, we have software which take care of all those small things. That's why I'm telling it's no more a very, it's no more a difficult task for the researchers to write an article. There are software, there are artificial tools which take care of those small things. Okay. I just want to know, do you have any problem with respect to finding out a research gap? My question, are you all convinced with my point or not? That research gap is very important or a very significant point in submitting.
Sir, yes, sir, we are with you, sir.
Yeah, thank you so much, sir. Thank you. So I just want to take another example because that giraffe may not be outdated for all of us, and we are in a era of genomics and proteomics. So let me take one more example here for this. I'm taking one example from microbiology, tuberculosis, drug resistance. This is the research area of the research study area. How to make a proposal on this tuberculosis drug resistance. Suppose you want to write just a proposal how you can prepare a proposal on the tuberculosis drug resistance. You can follow a similar procedure to prepare proposal on any topic. See, sir, preparing proposal is really not a big task. We can prepare the proposal, but ultimately the funding agency, they look into the expertise that we have in that particular field. Okay. So coming to the artist, sir. Sir, any question? Sir, any questions? I could not hear you, sir. Somebody is asking question. I don't know from which NRC. I think from NRC Kakinada. Sir, is time over? Sir, Srinivas sir, is time over? Okay. Sir, I could not hear from NRC Kakinada. There is a problem with your audio system.
Sir, we can continue for another.
Thank you so much, sir. Thank you so much, sir. Thank you. So coming to the tuberculosis drug resistance, here also we go and check the literature available in this tuberculosis drug resistance. I will explain probably in the next session how to find out the literature also with respect to a particular area of research. I will explain definitely on how we can extract the literature review from different websites. Okay. The first thing that you have to see, you have to read the articles, the recent articles in the particular field. Okay. So after reading the articles, you have to find out a gap. Okay. For example, in case of this tuberculosis, see, when a person who wishes to make a proposal on the tuberculosis drug resistance, he reviewed articles. After reviewing the articles, and he found that okay, drug resistance of microbacterium. Okay, so most of the studies, they focused on the proteomics, especially proteins that confer resistance to this particular microbe. So he reviewed all the literature available with this drug resistance in microbacterium, and he found that all studies, they focused on only the proteins that confers the drug resistance to this microbacterium. So after that he understands, okay, so studies are only focusing on the proteomics. There are many things which may be involving in conferring this resistance to the bacterium. So those areas are also need to be studied to have a comprehensive understanding of the resistance mechanism of the drug against the drugs by the tuberculosis. For example, if you take the identification of gaps by this researcher, see, he identified a gap in what you know. See, there is not enough information about the genomic regulation of the drug resistance, which means, for example, the difference expression of the genes between the resistant and sensitive bacteria or their regulation mechanism. So he has studied some almost 200, 300 recently published papers, and all the 200, 300 recently published papers focused on only the proteins. Proteins play a role in conferring the drug resistance and all. And there are no studies that explains how the genes expression changes between the resistant bacteria and the sensitive bacteria against the drugs. Okay, so that is the gap there. If you find out the genes, definitely that the information will enhance our understanding of what is the current knowledge that we have with respect to this drug resistance. The current knowledge is that proteins, they play a vital role in drug resistance, and the knowledge that we are adding to this is that genes, some genes which are very crucial for providing resistance to the bacteria against the drugs. Okay, and some may look into even the regulation of these genes. How those genes which are coding the proteins that confer the resistance against these drugs, So the genetic regulation of those genes, if we could study, so definitely we can find out the way to make the resistant bacteria into a sensitive bacteria, so that tuberculosis can be treated very well with the currently available drugs. Okay. So finally, what we have to write the emphasis on the significance of this study. We have to emphasize on the significance of this study. What is our emphasis here? See, you want to learn more about the genomics of bacteria, because understanding this is essential to understanding the drug resistance. That means not only the protein role with respect to the drug resistance, but also the role of genomes with respect to the studying the resistance in the bacteria is required. So that you have to mention that, yes, because why it is important, why genomics is important apart from this proteomics. So you have to write, yes, there is a reason, because this genomics information will enable us or will help us to understand the process of drug resistance in a comprehensive way or in a completely. Got it, sir, my point? So not just writing the gap, sometimes you may find a gap, but that gap may not be a significant one. In such case, so no one will care about it. Some people will come, sir, yes, we have identified gap and we want to work it, but please keep it in mind that, so that the knowledge that you are filling in this gap, whatever gap means, that means there is no information, and you have to fill that gap with the new information, so that you will get a whole idea about that particular process. So whatever the information that you are filling there, that should have a significance. If it doesn't have much significance, you are just filling some information there, so definitely that will not be appreciated by the research community when they review your article, or a granting agency when they review your proposal. So I want to take a break here, because many things are there to discuss or left over for discussion. So it's not possible to cover in the next 10 minutes. Even if I move into the next section, which will be not completed in the next 10 minutes. So I request the Srinivas sir to wind up the session now. And if time permits, in the next 2 days, we will move to the we will discuss the remaining aspects of the scientific writing, or writing of the research article. Our participants, if you have any doubts up to this section, you can ask me.
So the presentation is open for discussion. The participants over the analysis of the ecology state are requested to interact with the research person. That was like
Close this and close
Rajsekhar sir.
Yes, sir.
When after reviewing so many articles, we got a idea like a research gap, but we don't have proper materials and methodologies. Then what we are going to do, sir?
Sir, please ask once again.
We can identify the research gap or any problem based after reviewing so many articles.
Sir, you will not find such a situation. Definitely.
Sometimes the methodology is sometimes lacking, sir.
No, you will not encounter such a situation. You will not find such a situation, sir. Definitely, when you read the literature, you will find methods for conducting your research. You will not encounter such a issue.
Most of the methodologies, the problem is same. Methodologies are mostly same. But if problem is new, we need to find out new methodology, na sir.
Sir, see, finally the issue is that you have to find out an answer for your research question. So different people may have a different approaches in solving the problem. So it need not to be same with all. See, we can't say that everyone should use the same method to find out answer. We cannot demand that because different people may have a different options for finding the situation. For example, if you want to study, if you want to identify, for example, let me say, if you want to identify a gene which is responsible for a particular phenotype, see some scientists they take the help of the overexpression studies. Some scientists they use the gene silencing studies. Ultimately, they both identify the gene which is responsible for that particular phenotype. So different people follow different approaches to find the answers depending on the instruments available with them or depending on the grants they have with them. So all these matters while conducting the research. Sir, are there any questions from participants? If no questions, if there are no questions, we can wind up this.
An interaction from NRC Kakinada.
Sir, good afternoon, sir. Last second, sir.
Yes, ma'am. Yes.
This is Dr. Kiran, lecturer from Zoology and NRC Kakinada, sir.
Sir, your audio is not clear, sir. Please excuse me. Your audio is not clear.
Dr. Kiran Kumar, sir.
Yes, sir.
Yes,
sir. Yes, sir. Please ask the question.
What is the difference between research article and research publication?
Sir, your question about research article and publication?
What is the difference between research article and research publication?
What is the difference between the research article and publications? As per my knowledge, there is no difference. Maybe there may be some difference. If you feel, if you answer that, really that will help me to know the.
Sir, can I answer it, sir?
Sir?
Sir, can I answer it, sir? Can I give answer for that, sir?
Sir, I don't know the difference between these two. Even in the beginning, I said I have very little knowledge about it. Whatever knowledge. Sir? What, sir?
Answer about sir.
Yes, sir. That's what I'm asking you. Please answer that one so that everyone could.
Research publication.
About it.
Research article is a original research work which is published in journals. And research paper is also a personal research work which is presented in the form of a paper or a poster or a symposium.
Okay, rather than having this confusion, let's confine to one word, that is research publication. Both are publications. Are you agree with this, sir?
Both doesn't come under publications.
Both are publications, research publications. Okay, right, sir. Thank you, sir. Thank you so much.
I request NRC Kakinada to propose NRC Anantapur to propose vote of thanks on behalf of all the NRCs and all the participants. Vote to NRC Anantapur.
Myself, Jayapal. So I'm very much delighted to thanks Australia. They gave an elaborate lecture or elaborate presentation on how to write a scientific paper. Actually, we did a lot of work to prepare the Excel sheet almost in 6 days. So you understand that Excel sheet easily can write your research into a publication or a concept paper. So on behalf of all NRCs, I would like to say thanks to the NRC once again. So on all the participants, on behalf of Algebra NRC, we are very much thankful to you all. Thank you, sir.
Thank you, sir. Sir.
Also, I must say thanks to the commissioner, sir, and other officials who are over there Also the Srinivasan sir, especially for making this session happened. So thank you so much, sir, and other participants who have patiently listened to me. Thank you so much. Thanks to all of you.
Small announcement to all the NRCs. Afternoon session will be it, that is around 2:10 we will join. 2:10.
Okay, sir. Okay, thank you so much.
సార్ 2:30కి అన్నారు సార్ జాయిన్ అయ్యారు ఆయన జాయిన్ అయ్యారు జస్ట్ నేను మాట్లాడాను సార్ గుడ్ ఆఫ్టర్నూన్ సార్ ప్రొఫెసర్ నియాజ్ అహ్మద్ సార్ వెరీ గుడ్ ఆఫ్టర్నూన్ సార్ హలో
Can you hear me, please?
sir your audio is very much low sir
Can you hear me, please?
please check the audio once
Yes, sir. Yes, sir. Oh, great.
Hear me, please.
Madam,
All of you, please.
check the audio once
Yes, sir. Yes, sir.
oh, great.
Please.
Raju, sir.
Madam,
hello
you, please.
Madam, వచ్చేయండి ఇక్కడికి, వచ్చేయండి.
Yes, sir. Yes, sir.
నేను ఉంటేనే
anybody from any NRC please respond sir audio is audible to you
Hello, sir.
yes
NRC DKW responding, sir.
NRC Nellore audio is okay madam
Sir, NRC main Anandpur is responding, sir.
NRC తనకు రెస్పాండింగ్ సార్
yes sir
Srinivas, sir, shall I
Yes, ma'am.
introduce, sir?
over to NRC Nellore I request to
NRC Srikakulam response, sir.
hello
Yes, sir. Yes, sir. Responding, sir. Thank you, sir. Thank
I
you,
request Dr. Sriranjani madam to introduce this session resource person, Professor Niyaz Ahmad sir, the participants across the state from various NRCs. Over to NRC Nellore.
Thank you, sir. It is with great privilege that NRC DKW Autonomous Nellore got a wonderful opportunity to introduce to you all distinguished Professor Niyaz Ahmad sir, who embodies the spirit of academic excellence and dedication to learning. Professor Niyaz Ahmad sir stands as a beacon of inspiration for all of us and those embarking on their scholarly journey. Professor Niyaz Ahmad sir is One Health epidemiologist and professor of biotechnology and bioinformatics at the School of Life Sciences. His illustrious career is adorned with the prestigious accolades, including the distinguished and coveted India's foremost science prize in the area of medical science in 2016, Shanti Swarup Bhatnagar Prize for Science and Technology, for Sir's contribution on pioneering studies in the area of molecular epidemiology in India. With a formidable reputation built over the years of groundbreaking research and scholarly contributions, Professor Ahmad sir stands as a beacon of knowledge and innovation. Sir's scholarly prowess is evident in his extensive body of work comprising numerous groundbreaking publications in esteemed scientific journals. Sir's research spanning infectious diseases, molecular biology, and genomics has significantly advanced our understanding of microbial pathogenesis and therapeutic interventions. Sir also served as the Senior Director at the International Center for Diarrheal Disease Research, Bangladesh, in an international leadership position from 2016 to 2020, March 2020. Sir is Fellow of American Academy of Microbiology, the Royal Society of Chemistry, UK, and Royal Society of Biology, UK, and the National Academy of Science, India. Professor Ahmad sir serves as the founder editor-in-chief of Gut Pathogens Journal since 2008. In 2017, Sir was awarded the Microsoft Azure for Research Award in Public Health to work towards genomics of identification of new genes and novel pathways, underlining the multiple drug resistance phenotypes in bacteria. Sir has earlier received the University of Hyderabad Chancellor Award from then Chancellor of the University, Dr. C. Rangarajan, the former Governor of State of Andhra Pradesh and Governor of Reserve Bank of India. Sir is also the recipient of National Bioscience Award of the Government of India. And Sir holds appointment as Adjunct Professor of the Academy of Scientific and Innovative Research. He is the corresponding fellow of the European Helicobacter and Microbiota Study Group in 2011. Sir research main interest is about how bacteria interact with humans and the environment in order to evolve fitness advantages such as multiple drug resistant and strain host specific virulence. Sir work provided wheels to the emerging discipline of functional molecular infection epidemiology. Please join me in extending a warm welcome to Professor Niaz Ahmad sir in today's session, whose passion for teaching and unwavering dedication to scholarship will undoubtedly leave an indelible mark on our academic community. We welcome you, sir, to this session. Thank you, sir, for this opportunity.
Thank you so much. So, speaking after the lunch actually is little tricky because I also have a job to keep you awake. So, I hope I will do that. Please be here. I need your ears for the next 45 minutes or so. And if you have any burning questions, you can ask, but I would like all the questions to be asked at the end of the talk. Are you able to hear me? First of all, you tell me.
Yes,
And
sir.
also, are you seeing the slide? Can the do you see
Yes,
the
sir.
slides changing?
Yes, sir. Yes, sir.
Okay.
Yes, sir, it is.
Okay.
No, sir. Slides are not changing, sir. Only the
Okay.
slide is
Slides
changing,
are not changing, sir.
Okay, we will try. Don't worry. Are they changing now?
Yes, sir.
Yes, sir. Now it is visible, sir.
Okay, so you know the title of the talk is about evolution and adaptation. And this is the same thing that I talked earlier also during the HRDC seminar here in the University of Hyderabad, where most of you were present or many of you were present. Okay, so this talk is about to understand the core concepts of evolution from a bacterial organism to higher organisms such as human beings, you know. And the idea of this talk is because you people are also involved in teaching to undergraduate students. And many of our own students in the classes also ask some questions about evolution and the theory of evolution and the synthesis of evolution. And some of these questions are really tricky. So under the new education policy, we have enough of curriculum, which is full of, ecology, evolution, genomics, and, modern concepts in biochemistry, synthetic biology, and so on and so forth. So I think as a teacher, we all must also understand different viewpoints and, dynamics of the evolving discourse around evolution and the core concepts under evolution. Are you seeing it now? You are with me. If you are not, then you tell me. Okay. So the core concepts under Darwin's theory are to be understood also in light of the integrated synthesis. Okay. It has not to be seen, or should not be seen only through the inherit or the adaptation, natural selection. But there are, there is a modern synthesis and there's an integrated synthesis, and the concept is here on your screen. It has a scope for all the things to be put in the context and try to understand the process of evolution in light of some of these findings, that are related to classical genetics, the neutral theory, to epigenetics, to natural selection, to genomics, to plasticity, variation. And all these concepts are quite a lot of established concepts that also are part of the curricula here and there. Okay, so two things they emerge out of Darwin's theory. Species are not immutable, but change or adapt over time. And natural selection is the driver of that change or adaptation. Okay. Now, going forward, so there are some of the major challenges to the evolutionary theory, including Darwinism. There are major challenges, there are major questions that are still unanswered. And because of these challenges, there is an skepticism. And there is also a debate in some of the countries whether the Darwinian evolution should be part of the curriculum in the schools and all. And we say, yes, it should be, and we must allow a detailed discussion around some of these basic concepts in order to also understand or evolve the higher order narratives, and that is where the science progresses. I mean, we should not be at that level of fanatics to not discuss the core concepts in evolution from a Darwinian perspective, but we should also not shy away from some of the questions that need answers. And the answers so far are inconclusive. And this is the nature of science, and the synthesis of science, that it doesn't stop. The answers emerge incrementally, but they need a lot of empirical evidence and explanation, and the explanation should be also satisfactory. But currently, as it goes, that we are struggling, or the sort of the state of art currently is struggling to find some of the answer to these solutions, such as these questions, such as the mind-boggling complexity of the universe and the biological systems amidst perfect balances and sinks. Okay, are the slides changing now? I'm not getting any answers.
Yes, sir. Changing, sir. Changing,
Okay,
sir.
great. Another question is how life at the first instance originated on the Earth. It's a big challenge to crack. And then the irreducible complexity of some of the molecular machines, they are thought to be evolving over a period of time by bit acquisition of characters, and then the perfection of the functions under a selection pressure. But then there is an analogy, or there is a challenge to this, concept that these machines could have not been functional from the day one, because the organisms must have need their functions from the day one for them to survive. So if they were to arrange over the millennia in a bit by bit fashion, then how the first organism could have coped the challenges of life. Then there is an absence of fossil records, for example, in telling forerunners and ancestors of some of the or billions of species. They call it as Cambrian explosion. And the cognitive evolution, the evolution of speech and sophistication of human species so far, they appear to be very deviant from their so-called human-like ancestors. If we assume that there were ancestors which were human-like, then why they are not surviving today? What they are not around us still, if they at all were there, why they were not as excellent in cognitive terms and speech and narrative and storytelling and art and sophistication of human living the way that we are today. These are some of the questions that they themselves need a lot of lecturing. Each of these points may be a lecture of 2 hours. Okay, so keep these points in mind. We are discussing with an open perspective. But at the same time, I would like also to use some of your thought process to see, for example, these are some of the cellular processes that are, condensed in one slide. This is a chart, courtesy to Roche molecular systems, that they have developed this chart. And in good olden days, not so olden days, when I was a student, we used to see this kind of in the office rooms of our professors of biochemistry and all. And I got a motivation from those days, that these charts were still in my memory. And I try to ask myself the questions as to how these complex biochemical reactions in the form of a very, very efficient integrated synthesis could have automatically evolved. If this is not a random evolution, could that be a guided evolution? Because these processes follow a lot of rules and a lot of systems. For example, never at a given point of time the biological homeostasis goes astray, and be defiant of the natural laws. For example, the laws of the chemical bonding, the laws related to the codon usage, the laws related to the energy, thermodynamics, entropy, enthalpy, the laws related to biochemical synthesis and phosphorylation and so on and so forth. So all these laws are basically grounded in the functioning of these complex pathways. So that means they very well follow the laws that are set forth. So what are the laws in the universe? For example, there is a law, there's a concept of relativity, there's a law of thermodynamics, the laws related to the synthesis of, biochemical synthesis, the bonding, the codon usage, and, so on and so forth. So then when these laws came into existence, then who made these laws? If we say that all molecules and synthesis of life, they follow the rules and laws of nature, then how the laws at the first place came into existence? That itself is a question, So these laws are very well there, and biological processes do not, supersede the brackets of these laws of, natural, sustenance in the universe. The first thing that comes in mind, by the way, is that how the life into existence in the beginning when nothing was there. So something that would have been created from a scratch could have been a simple molecule, a molecule like a nucleic acid, for example, which are the building blocks of life. But then how? If DNA had to come to existence, then the DNA, must be synthesized, and then the DNA must replicate itself. So for replication of the nucleic acid, the proteins will be required. So when nucleic acid was not there at the first place, then how the protein will be present? So these are some of the things. And if protein were to be there, then the amino acids were to be there. And these precursor molecules, how they have come. Then, the most fashionable theory to explain this in today's, scenario is the RNA world hypothesis, which is also struggling, to prove itself. There are lots of arguments about the tenability of such a theory, which we will discuss, Then we will discuss them in an academic sense. I will try to ask questions as to how it is possible for the RNA to be the genetic material since the beginning, or to be the prebiotic, initiation point of the world and the life and its hierarchies. And the argument here is that the RNA may have been too sticky to have served as the genetic molecule of life in its own. So the tendency of nucleic acids to self-hybridize begs the question of how these molecules were separated to facilitate replication. Okay, because they must, RNA structures are too sticky. So that means to separate the RNA hairpin structures, the temperatures must be raised to very high levels. So under such a high level of temperature, how it is possible for any protein to function, for that matter, if the replication were to occur? The question, and there is an answer that there could be the, geothermal situations under the sea, where the hot water, lagoons were there, the accumulations of hot water could have facilitated this and that. So there are explanations, which may sometimes sound, a lot of oversimplification or just a fairy tale. But then if we assume that, okay, let that be that there were hot springs or something where the life originated, where the RNA could be straightened, and made less sticky so that it can, then how peptides which might have carried out transcription and translation could have essentially created themselves in an RNA world? That itself is a question. And then how heterogeneous bases could have formed. And there is a concept here, you see the cartoon on your screens on the slide is from a PNS paper, I have taken, and there is a concept that interplanetary dust particles could have had these already certain nitrogenous compounds. They may be the precursors of the nitrogenous bases. So there is a lots of imagination. It is open to question, and the questions are many, about the beginning of the life itself, like how the life originated on Earth. That itself has lots of questions that are emerging, and the theory of RNA world hypothesis basically struggles very badly in light of these points. So consider these points. I beg your patience, because this is kind of very verbose slide, but all points are very relevant. So stability of RNA molecules, we discussed this, that how they could be stable. They are less stable as compared to DNA, and therefore making it very difficult to sustain the necessary conditions of RNA-based life to emerge and persist over a long period of time. Then the prebiotic synthesis of RNA, the spontaneous formation of RNA molecules under prebiotic conditions, remains a significant hurdle for that matter. Are you with me? Okay, so it remains a significant hurdle. Let me see whether I can the slides. Okay, they are going. And then there was also a possibility, or there could be a difficulty that under prebiotic conditions, the chemical reactions required for RNA synthesis are too complex, and they may have not occurred readily in early Earth-like environments. And that there's a difficulty of self-replication with long RNA molecules. Then catalysis problems, for example, while some RNA molecules could have a catalytic property owing to them being called as ribosomes, their efficiency and versatility may not be sufficient to support the full spectrum of cellular processes. Then the competition with other molecules, the RNA molecules would have had to compete with various other molecules for resources and chemical reactions on the early Earth. This raises questions about how RNA could have outcompeted the alternative molecules to become the foundation of early life. Then the evolution of complexity, like transitioning from simple RNA molecules to the complex cellular structures and functions seen in modern life, represents a significant evolutionary leap. Understanding how this complexity emerged from a presumably RNA-based world remains a major challenge. Then there were geochemical constraints. For example, in an RNA world scenario like the temperature, pH, availability of key elements, we are not able to standardize a reaction in the laboratory itself. Sometimes we struggle to standardize RT-PCR and other things. So can you imagine in a prebiotic condition on early Earth environment, lots of, meteor showers, then, unstable pH, there is a fluctuation of temperatures and so on and so forth, availability of a lot of dust particles, the interplanetary dust and so on and so forth, so many compounds, so many things around, how these reactions could have precisely happened. Some constraints are there, and the experimental validation is another big constraint. While laboratory experiments have demonstrated some aspects of RNA self-replication and catalysis under controlled conditions, replicating the entire process of RNA world emergence in the lab remains very elusive. So experimental validation of the key steps in RNA world scenarios is crucial for establishing its viability as a model for the origin of life. So you may have got my point of argument that this is simply not tenable over a period of time, as to how a particular stretch of nucleic acids or a stretch of nitrogenous bases or nucleotides will come in a very meaningful manner to serve both as a template as well as a protein or a catalysis trigger, and then retain its lifestyle and create a niche for itself and be very specific at the same time. This needs a lot of statistical support, which is currently not available. I mean, there is a lot of imagination, but there is a lot of difficulty in hand. Then going forward from here, has the system come just by chance or random guided interaction, trial or error? Then you have to consider a probability theory for that. It is obvious that probability of such a numerically ordered arrangement, for example, in the early RNA molecules, it decreases rapidly as the number of components increases. When the complexities, the specificities, and the desired direction of evolution in shaping the life or the early life and finding a niche for itself, under these circumstances, the probability of such an ordered arrangement becomes very minuscule. For any linear system of 100 components, for example, in a specified order, the probability is 1 in 100. So something of a higher order system, like requiring a high degree of order, such as a very specific stretch of RNA, with the probability is mind-boggling, like something like 10 to the power 600. And so whether this probability is supported in a time-space universe of our kind, in a time and space, I mean, this is not possible. And I will tell you why it is not possible going forward. But then imagine that such a situation, hypothetically, if at all it is there, but then a very small specific RNA life at the beginning and some of the pathways that it may have triggered could have not evolved, into such a sophisticated system, such as the human brain, for example. Now they take the take the example of human brain, and all these things in 2 to 3 billion years. That's it. In such a time, such an order of complexity, is not imaginable, or at least not supported by probability, and not by the resources and time space constraints of our universe. And now consider the brain. Human brain is a complex and compact structure, and it is the most complex organ that that could have evolved, or that could have aggregated or formed from the small simple precursors as RNAs we see, or the proteins or the peptides, and then gradually to such an excellence. So whether the things support it. I mean, the question our students also ask in the lab, that how tenable is this idea, like that it has come to this much sophistication in a few billion years, And so over 100 billion neurons and 60 trillion synapses that can be assumed to work like a transistor, and therefore the brain's processing power is approximately equal to more or less 33,000 iPhone processors. Can you imagine? How many of you have an iPhone? Can you raise your hands? So many of you, or many of you. So if not iPhone, maybe another phone, Android phone or whatever it is. The speed of the processor sometimes overwhelms you, and the power of the processor can allow you to do so much of fun with your mobile phone that all the time you are busy with it. And you see that how much the fun the brain can do with the 33,000 equivalent processors, capacity that the brain has. So our brain processes information in the range of 20 petaflops, which are the floating point operations per second that it can perform. However, the brain's true strength signifies in its power to parallel process the observations and signals and dynamically adapt to various tasks, something current computers cannot do. Imagine you are driving a car. Is there an involvement of brain in driving or not? It is. And driving a car, at the same, you are listening to music. At the same time, you are responding to the traffic signals. At the same time, you are negotiating with the other traffic. At the same time, you have your eyes on your speedometer. At the same time, you are using your gearbox and transition system of your car. At the same time, you are exercising control on your acceleration. At the same time, you are talking to your fellow passengers. At the same time, it is running in your brain how much time you have to arrive at your destination. So isn't that all a parallel processing that your brain is doing while you are driving? You are also processing a lot of information. You are getting into, communication. So there is a lot of observation, communication, lot of signaling, lot of coordination of actions, movement, and lot of decision making that is happening in a very, purposeful, very productive direction where you are supposed to arrive in your time directly on your destination. So doesn't it overwhelm you how your brain is functioning? And it's not a matter of just one mutation. There is a recent study in 2022 that is published in Science, that delineates that human DNA contains about 20,000 genes, and about 96 of human specific mutations were found that could change the structure of a protein in the brain, and one that altered a gene called TKTL1. Okay, so TKTL1 transketolase like 1 has been studied herein, and just see one mutation or one lysine, if it is replaced with arginine, then the basal radial ganglia in human brains, they enrich like anything, and there are billions of neurons, whereas neurons were not that replete or not that enriched in the Neanderthals, for example. And that signifies that how Neanderthals were not enjoying that level of cognitive sophistication and excellence that we enjoy today. Understood? So it's only a matter of just one amino acid being different in humans, modern humans, then. So whether this was a purposeful substitution or whether this substitution has occurred just by chance. And how beautiful chance it would have been that it has created such a sophisticated human system of cognitive excellence. Are you with me? But it is not, broadly speaking, it is not a matter of just one mutation. When you see the entire scenario, of how the brain or how the cognitive functions they come into existence, or in the higher organisms such as the human beings of the today, the TKTL or NOVA1 or PPP or purine and lipid biosynthesis, all systems are connected. And for all these things to connect in a very favorable manner, it needs a lot of probabilistic support. It needs a lot of statistical support, for it to justly happen. For it to happen in a very purposeful evolution, in a very guided manner. So if at all it is happening through evolution and adaptation in a guided way, then so it be. But it cannot be random. If we assume it to be very random, then this randomness has to be justified, on the basis of probability. On mathematical terms, it should be justified, because the constraints of our time space in the universe and the time that we have from the first life to this much cognitive excellence is not unlimited. It is a few billion years. And whether that much time actually explains or signifies this level of sophistication that has emerged. So in light of this, consider these points. Cognitive development appears to be a complex phenomenon, not mediated by certain single gene functions. So what about epigenetics? It's possible that several common genes could have differential expression or could be suppressed by epigenetic means. The chance and probability do not support successive acquisition and accumulation of mutational advantage only in one species all the time. So if this was to happen, why it has happened only to the humans? Why this could have happened in all the hominids or in all other species, including animal species? Only intelligence and parallel processing capabilities are not about being the human being. What is being human? I will tell you in the next slide what it makes to be a human. So most mutations are partly or wholly deleterious, and natural selection could possibly serve to reduce the deterioration of life, not evolve life to greater levels of complexity or create further complex forms in hierarchies. Consider this statement. And human and chimp Y chromosomes, for example, are horrendously different from each other. So how these big differences are there, and how a lot of body form overlap and lot of overlap of system, yet certain very critical differences leading to very high level of cognitive excellence or sophistication of functions. So these are some of the points we must consider alongside when we are considering that if it could be an outcome of a random vagabond process of evolution and adaptation under natural selection. Are you with me? Just say one word.
Sir, yes, sir.
Great. So we go forward from here and see what it makes to be a human being. Only intelligence, parallel processing of the signals, is not being human being over other ancestors or over other ancestral forms. If we call them as ancestors, or other hominid forms, if we call them to be the descendants of a common ancestor or whatever. So what a fully evolved human, which is, kind of a best of the stature and best of the body forms, is all about. So beauty, wisdom, elegance, speech, narrative, creativity, cognitive excellence, quest, curiosity, memory, number games, craft, mercy, kindness, pride, gratitude, repentance, moral and emotional superiority, spirituality, language skills, and expressional sophistication. So why these things are only enriching in the modern humans? Whether the theory of evolution and adaptation by natural selection could shape these features very exclusive in making a human personality, in all kindness, sophistication, beauty and elegance, and legacy, and language and expressional sophistication, how it can come into existence. If we say mercy, kindness, pride, gratitude, repentance, are there any biochemical processes in the backbone of these properties? If they be, then can our evolutionary time span and the time, space, resources of our universe support, evolution of this beauty of human character, which is best of the stature? Keep this in mind and we move forward. If you say that this has come by chance, this has come randomly, then it will be like, I should give you an example of, which is, I have a citation here. This gentleman has cited it, that could infinite number of chimpanzees on typewriters for an infinite length of time type the complete works of Shakespeare. There exists 26 letters, 10 numbers, 4 punctuation marks, and a space button. So totally there are 41 options. So utilizing the factorial 41, this number is an immense figure, 3.3452527 into 10 to the power 49 or whatever, an immense figure that exponentially grows when considering Shakespeare's vocabulary. So the Shakespeare's vocabulary was of 30,000 words. So what is the likelihood that one of these monkeys, even if they are allowed, for example, 4.5 million years, they say that the human life or human-like life forms or life on the earth is for something like 4.5 million years. So if this much time also is allowed, the task of achieving this, like some monkey will one day type the sonnet of Shakespeare, becomes astronomically very, very small and minuscule, not to mention his entire collection. The task of achieving this is so daunting that the concept of infinity may not be sufficient, that the galaxies will exhaust themselves before producing a Shakespearean sonnet from one of these random sequences of the strokes that the monkeys will type on the typewriter. So you also consider this, that something cannot come just like that by trial and error, by randomness, by allowing a chance. Also whether this chance occurrence has anything to do with the reality of the concept of probability, or reality whereby our time space in the universe could support this to occur, whether our galaxies could support in terms of time and space for this thing to happen. Why go that far? Why, human brain is such a complicated thing, just go inside the cell, and inside a human cell, consider the nucleus and consider the nucleopore complex. Very small thing. In the human body, there are cells. In the cells, there are nucleus. In the nucleus, there is a nucleopore complex. And, this is a kind of import-export. The small organelle or a small thing that actually is controlling the import and export of the macromolecules, inside the nucleus and out. So the nucleopore complex is composed of approximately 34 different nucleoporins, are the proteins that is irreducibly complex nanostructure of an extremely vital housekeeping significance. So had this not been functional from the day one, how the cell itself as a unicellular life even to assume, how it was functioning. Bacterial systems may be very simple. Their protein synthesis and their other things are very simple. Still, they are very dependent on other biomolecules. But if you see that a complex life form in the form of a oligocellular creature or a small early body forms, how they would have been functional if the nucleopore complex was not properly organized, or if one of the 34 nucleopore proteins were missing. That implies that this could have been present since the beginning in its fullest of the form. If it were to evolve over a millennia, then how life would have sustained until then, that point of time. There are arguments also. I have cited the papers here with due regards and respect to the works that have been cited. People are of the opinion that the rudimentary forms of these things could have been existing, So it is easy to say that, okay, there could be simpler forms, there could be rudimentary forms, there could be other identical molecules around which could have been co-opted or which would have been, recruited, and there could be umpteen number of explanations. But then is it possible, if we ask, it's not possible for such a sophistication to be just coming out of nowhere or out of only random events. So why can't then we also accommodate the alternative viewpoint that they were present since the beginning, or they were the first to become active or functional, because they are very, very essential housekeeping things that were to be present. For example, why go to the human cellular system in bacteria, for example? As I told you that the bacteria are much simpler in terms of biochemical functions, in terms of their metabolism, in terms of their information processing, in terms of their survival, then they also have a lot of complicated structures that they have evolved. So I will talk about the bacterial flagellar apparatus, for example, and the bacterial flagellar protein have, so many, like more than 40, 50 small components that must come together for the flagellar motor to be functional. And I should tell you why flagella is very important in a bacterial system. If they do not, flagella is kind of also a virulence factor in bacteria such as Helicobacter pylori, on which we work quite a lot, and typhoid organisms and other organisms, they provide them the ability to be motile so that they can move and change their niche. They can move away from their predators. They can move to the site where they can get the food. Okay. So when you culture these bacteria on a Petri plate on artificial media, sometimes you see that the flagella do not express, and the motile bacteria, they become static, and spiral bacteria, they become cocoid. So there the flagella is not there on the Petri plate. Why? Because it doesn't need, because that niche is not for it to swim or for it to be motile. Some are still motile, so that their colonies move. So this bacterial flagellar apparatus is required for them to provide a survival advantage, so that they can scavenge for food, they can move away from the predators, and they can also go to their desired niche. For example, there's a lot of thick mucus on the gastric epithelium, and these bacteria must swim the mucus out in order to cling to the epithelial cell surfaces. And for that, they need the flagella. That means flagella is a vital housekeeping function that provides a unique survival advantage to this bacterium. Those bacteria who are deficient in flagella, they cannot survive, they cannot enjoy this advantage, and therefore they could as well be outcompeted by other actively motile organisms. So they would have not survived had there been a single, misplacement in the assembly of flagella. And that is how we say that flagellar apparatus in bacteria is a indispensable and irreducibly complex system that could have seemingly not evolved through random successive mutations and adaptations under natural conditions in a bit by bit manner. Rather, it can be assumed that they were present or they were acquired en bloc. Of course, there are arguments, there are people are providing incremental evidences that there is a possibility that they may acquire similar molecules through horizontal gene transfer, through recombinational events from their vicinity, or they recruit or co-opt, molecules with similar functions or structure, and there is a lot of adaptation and flexibility there. But again, this has to be supported. We are not negating that argument at this stage, but there should be also possibility to consider that if they were present right in the beginning. For example, the pathogenicity island in Helicobacter pylori, called CAG pathogenicity island, it appears to have been acquired in block in bacterium, because this is such a complex structure that it cannot be imagined to have assembled bit by bit over a period of time. If they were to assemble, then the 50,000 years of presence of Helicobacter and association with the humans or human-like hominid forms could have not been justified. This could have not been justified in that much period, like 50 to 60,000 years or 100,000 of Helicobacter. Helicobacter's association with the humans or the human-like body forms has been described to be since the out of Africa hypothesis, out of Africa bottleneck, and so it traces back until that time. So 60,000 to 100,000 years is not sufficient for a very complex structure like the CAG pathogenicity island, which is a type 4 system, to have evolved over a period of time in a bit by bit fashion through recombinational events, or through synthesis or whatever. You see the pilus formation, or the arrangement of the barrel-like or a syringe-like structure of this virulence factor, the pathogenicity island of KAG type in Helicobacter needs a arrangement of a lot of protein. It's a mind-boggling structure, but it is very, very necessary for the survival of the bacterium. And why it is necessary? Because this whole barrel-like structure must be used, must be used to eject out a virulence factor called KAG protein, the cytotoxin associated gene A encoded protein, which is a signaling protein or a microbial oncoprotein that must be injected into the human host cell for the signaling or inflammatory signaling to start. And you know what inflammation gives the bacteria basically? The inflammation provides the bacterium a sustenance, a livelihood, a source of food, a carbon source, for example. The inflammation would trigger, the mucosal exudates. And the mucosal exudates are rich in mucosal sugars. And that is where the bacteria are looking to use such mucosal sugars as the carbon source. And that is the modus operandi, behind the pro-inflammatory signaling of the type of KAG, for example. And such a sophisticated, engineered, horrendously complex system should be intact if the bacteria will have to take advantage of the inflammatory processes for their survival. If this system is not functional, if the KAG protein is not able to come out of this barrel-like structure, or if this barrel or syringe-like structure is deficient in some of the component parts, then the inflammation and triggering of the mucosal sugars and everything for the bacteria to survive would have not been possible. And this bacterium would have never survived that long. It is today, if you say 50,000 years or 100,000 years that it has been associated with human-like body forms, then this should have been there all the time. If we say that it bit by bit over the millennia or in billion years, then this does not justify. The bacteria would have long been outcompeted. It would have not survived. And we say, but there is a possibility to say that there would have been recombinational events such that only those bacteria which could successfully generate these complex structures and carry them forward could have only survived. But we see several bacteria which are deficient in this system. This system, as I told you, has been acquired in the genome or in their chromosome through the horizontal gene transfer mediated acquisition of pathogenicity islands, which is an en bloc acquisition, not a bit by bit acquisition. And this is a larger, spontaneous event that happens. And Darwin did not thought of this, and he didn't explain it. Had he thought of this, he would have explained this, but the thing doesn't support it. I hope you are getting my point. By seeing this, nice picture, survival of these pathogenic forms of bacteria by virtue of their specific virulence factors, which are very complicated in organization, and therefore it cannot be assumed that they would have been, shaping themselves over a period of time of millennia or billions of years. There could also be a possibility that these systems might be very well existing since the beginning. A similar system in Helicobacter that we researched in our laboratory, was about a protein of Helicobacter pylori called HP0421, the cholesterol alpha glucosyl transferase. When we mutated this protein and knocked out of Helicobacter, you see in the center here the gel image of the cell wall, that the cell wall of Helicobacter collapsed, where the deletion mutant of HP421 was analyzed with respect to the cell wall integrity. That means it lost the flagella, it lost the spiral structure, it became cocoid, and its cell wall became porous, or its integrity was lost. At the same time, this pathogen also, which was knocked out of HP0421, became less efficient in biofilm formation. Otherwise, the wild type was able to form the biofilms. So when we replace this back, then the substituted recombinant of the HP0421 in Helicobacter regained all the functions. It retained the cell wall integrity was, put back to function. It became a good biofilm former, and it also became resistant back again to antibiotics to which it was sensitive earlier. You know, to which it was sensitive when the knockout was generated. You know, the antibiotics to which it was resistant, you know, it became sensitive when 421 was knocked down. That means the Helicobacter cell wall actually could give way to the antibiotics to enter and kill the organism. And when this was put back again, the sensitive forms again reverted back to resistant forms. So one outer membrane proteins out of 60 to 70 such different outer membrane proteins in Helicobacter could have such a indispensable character in cell wall biosynthesis, such a irreducible complexity of the system that when it was knocked down, the entire cellular envelope, of the Helicobacter collapsed. And so many derangements have happened. When it was put back, then it came to normal functioning, including the normal spiral shape was regained. So it can be also proved in the laboratory in a way in a model system like Helicobacter. That is the message from here. And you see that the spiral forms were, restored when this gene was put back again, and its, patency, or it's allowing the Ethereum bromide, for example, to enter inside, or antibiotics to enter inside, were again restored back. So it was impervious to ethidium bromide when the gene was replaced back, but it became, permissive to ethidium bromide as well as other molecules such as antibiotics when this gene was removed. So that means this gene is very important and significant in the cell wall biogenesis of Helicobacter pylori. So we thought, why not to consider it then as a drug target? That if you design a drug and target this protein somehow, then this protein can be blocked, and otherwise resistant Helicobacter could become sensitive to the mainline drugs or mainstream drugs that are usually given to control Helicobacter infections. Although these drugs may not be very efficient in controlling the infection, but then therefore this protein like 421 could be a drug target. That is what we tried, to make or suggest certain molecules through, computational chemical discovery. And this paper has been published, whereby the legend has been suggested, for this kind of a drug to come into existence. People can take this forward. But this works on the something like this model, which was published in Nature some time back, whereby the NDM1 NDM1 encoding resistant Enterobacter species, which were kind of super bugs of their, when they were administered with meropenem drug, they resisted it. And the cell wall could not allow, for example, the drug to enter inside. But when something like aspergillomarasmine was administered together, then the meropenem could act very well, could enter and act very well, because the aspergillomarasmine actually quenched the or removed the water molecule, and it could bind to the zinc ions, and therefore it may allow the meropenem to be functional rather than degraded, and that is how the sensitive bacterium is created, which killed easily by the other drugs. So this kind of a regime could be possible also in this case, where a drug can be targeted to this receptor HP0421 or a drug target here, and the legend can be very well developed through chemical synthesis. This is just an example, But then drug resistance is coming overwhelmingly to many different antibiotics. So we have a E. coli bacteria that we worked upon, and we named this Na114, and this E. coli bacterium could resist to 14 different antibiotic classes. So why this overwhelming resistance is coming? And this resistance is coming apparently after 100 years of the discovery of the first antibiotic, that was penicillin. So they say that in 100 years, many things change. The world order changes, the economical systems, they change, the narratives, they change, so many things change over a period of 100 years. So 100 years is a good time to change the things. So then you see, I would like to take you 100 years back in time. The World War II was unfolding, about to unfold. There was a memory of World War I, and a lot of people have died in World War I. When the wars happen, the war injury, a lot of gunshot wounds, the amputations, the sepsis, gangrene, and so many handicapped people, so many deaths, so many burns. So how this would have been managed in the absence of antibiotics? So World War I was fought in a pre-antibiotic era. And by the time the World War II could start, the antibiotic penicillin was already ready. So I just give you this scenario that you imagine that penicillin as an antibiotic was a divine gift to mankind. So whether we have cherished it or it over a period of 100 years. And they say that in 100 years, it would have saved 200 million lives. So saving 200 million lives in 100 years is a miracle. So we say that penicillin was a miraculous drug. So what miracle has happened exactly? So this may be our last but one slide, but I want to ask you one more time. Are you still there? You are not sleeping. Can you confirm me?
Yes, sir. No, sir. We are listening.
Okay.
We are listening.
Great. So I will tell you, I like a lot of tale-telling. So I will tell you a tale of Alexander Fleming. He was not the Alexander the Great, but nevertheless a great scientist, And this great scientist is, famous for lot of great accidents that have happened with him. That person was working on lysozyme, and he was working to make lysozyme as a drug against bacteria. So he was definitely famous for culturing a lot of bacteria in his laboratory, and he was, it was told that he was not so organized. So once that he sneezed in his laboratory while working, and some of his product of sneeze fall into a plate, and the bacteria on that plate died of that. So he, sort of became very curious to know as to what was in the sneeze that has killed the bacterium. And that gave, the idea of lysozyme being present in the human mucus. So a particle of mucus has fallen out of his nose on the plate, So such was his accident of discovery of lysozyme. And in the backdrop of that discovery, another accident has happened. You remember the monkey theorem, the random monkey, infinite monkeys for an infinite time on infinite number of typewriters across 41 parameters. So don't let it go out of your mind. So what has happened in case of Fleming is that in the September of 1928, Fleming decided to go out for a holiday. Usually August, September is a holiday period, in the Europe. And, he used to work in England, and the temperatures there are very, very cold. So for the bacteria to grow just outside the incubator, the cold environment is not conducive. So, the environment must be something like 30 degrees. And so when I analyzed the weather data during September 1928, it came out that September of 1928, when he left for his holidays, was something like very warm September in decades, where bacteria could grow, So this was one happening. Note down. The second happening was when he went for his holidays, he left the window of his room open. By accident or by chance, the window left open. You see the window in the photo. And he left a petri plate with a culture on it, on the plate, on his table open. And he went for his holidays. Then a species of mold called Penicillium, species Penicillium notatum or Penicillium or whatever it is. Out of 350 such species, only that species that produces penicillin moved to that building and then went to that floor, then went to his office laboratory through that window, through that plate onto the culture. It has fallen on the culture, and both bacteria and the mold grew simultaneously because the temperature was very warm. And when the mold was growing, the mold actually released some of its metabolites and penicillin there, and the bacterial lawn was cleared. When Fleming came from his holidays, he saw that a zone of clearance has developed. And so what is the reason? and there was a mold there. And he could just like, aha, so this is my solution. And the World War II just didn't start by then. And there were only 10, 12 years in hand to go until the doses were available. And the 80 million people were to die in World War II. So what Fleming has written or said, we all know that chance, fortune, fate or destiny, call it what you will, has played considerable part in many of the great discovery in science. So the outstretched hand of opportunity, he called it as outstretched hand of opportunity. What is that hand? Who helped identify this antibiotic from all those accidents? People say that this is a serendipitous discovery. So serendipity has origins in the name Serandip. Serandip was the earlier name of Sri Lanka, when 3 princes of Sri Lanka, they left for a voyage or a journey, and all the time very pleasant accidents have happened with them, and all the time the outcomes of those happenings were very, very positive. So that is like winning the lottery every day. So sagacity plus n number of pleasant accidents leading to positive outcomes is equal to highly valued discovery. Just like the remote nodes of a complex network joining each other in a highly favorable manner. A matter of perspective. It is not a description of the objective world. So then we come back again to the same thing. Does probability support that? Whether the time space could allow that to happen. Whether probability supports you to win every day a lottery, a bumper. No, it doesn't. So does that mean it was a miracle to save 200 million lives during the World War II and afterwards until today's date? So this was like as if this was a gift of mankind of antibiotics, because otherwise it would have been, it should not be like the entire humanity is wiped out of the surface of the earth. Remember, the discovery of atom bomb also happened in that time, during that time, just before World War II. And what some people were doing on one hand and what this humble scientist was doing on the other hand, that's how, you see Hiroshima and Nagasaki on the one hand were, completely destroyed. And then on the other hand, the penicillin could save, 200 million lives. And isn't that mind boggling to you? So in his, memoirs or in his biography, his biographer could write that we can almost see the fingerprint of God pointed to the direction his career should take him. And I mean, we all should read this book, his biography, and see what all that has happened to him. There's so many chance accidents that have happened. Each one of us actually experience these chances or opportunities, but we do not pay attention to it. We say the seeds of discovery are all around us, but the roots of these discoveries take the shape of trees only in the brains of those who are prepared, who think, and perhaps who may be used for this kind of discovery because they are very resourceful people. They are very rich in their resources, whether intellectual resources or whether material resources or whether spiritual resources. So that means they are the people who are selected for these kind of discoveries, Otherwise, so the seeds of discovery are all around us. But do we think on that? Can we become a resourceful person of that sort, that we may be actually that person to go to a great millennial discovery that way? Okay, think on it. 100 years now, we are full circle after that discovery of penicillin. And you see the mortality data are already on your screen, that the antimicrobial resistance is now very well poised to kill, 20 million people per year up to 2040. And therefore, 200 millions of life are to be lost. So antibiotic saved 200 million lives, and antimicrobial resistance will kill 200 million people. Is there a miracle to take place again? Are we waiting for this kind of a millennial discovery that should take place after 100 years? But do we have the, kind of building blocks of that discovery? Is that discovery going to be in the form of a drug or in the form of a culture or a regimen? How that discovery will happen? how that miracle will happen. If we say that that miracle should happen, take place again, then we should change ourselves, because the miracles will not happen to those people who do not have desire to change themselves. So what should we change of ourselves? You see the rampant abuse of antibiotics that people are doing. The antibiotic residues abound. The antibiotics being fed in the ration of the animals. The antibiotics being taken over the counter. So for 100 years, this abuse is going on. So unless we stop this, there is no miracle that is going to happen. And we do not see any such drug in the pipeline that may yet again revolutionize the scenario and saves these 200 million lives. Thank you so much, and I stop here.
Participants across the various NRCs are requested to interact with resource person, Professor Niyaz Ahmad sir. Yes, Madam NRC Nellore, proceed, Madam.
Yes, sir.
Did I take a lot of time of you?
No, sir, it is all right.
Sir, any questions from the participants?
Looks like participants do not want to ask any question. They have understood everything. When you go to the classroom, your students might ask you some questions, so you should be ready with the answers.
Good afternoon, sir.
Yes, good afternoon.
Sir, question is like in earlier times, if we take the census of India or profit number, I think the computer skills were very well developed and they used to, receive signals and they have written whatever they have because of computer skills. Now we have come to a stage where most of us are with less computer skills and just human. And just a few weeks ago, we lecturers have been trained to use artificial intelligence where we can just, record voice and the artificial intelligence is going to do everything. It is going to prepare our slides and in our voice it is going to, take the class also. So I'm scared that I'll be using my cognitive skills further on creativity also. I'll be using. What do you say about this, sir?
So, your voice was not clear, but let me tell you what I understood is that you want to say that in good olden days, people used to do a lot of brainy works. The brain used to work quite a lot. And in today's world, we are less using our cognitive skills and more dependent on the manuals and gadgets and, programs and sophistication like AI. So is that the question?
Yes, sir.
Yeah, you cannot stop the wheels of revolution. They are changing. We are all moving forward with the revolution of time, with new things coming to our assistance, and there is you have to adapt and evolve. And that is what is the beauty and flexibility of the human system. Now everybody is trying to use these things, and people earlier were used to struggle even to find out some common things from Google and all. Now people AI and other things and chat GPT and so on and so forth. Ordinary people are doing AI inspired searches rather than simple searches. So people try and adapt and evolve to the system. Now we are doing lots of online transaction. Earlier people were struggling to do a transaction in the bank, which was simply filling a check and giving there and taking the money and transferring the money by applying in the bank, and people used to struggle. Sometimes they used to be in the queue for hours, and a whole day used to be wasted in transacting in the banks. Now people are transacting at the press of a fingertip, just in a jiffy. So that means they have evolved, they have learned. And so the human learning is a continuous process. There is no second opinion about that. That's the beauty of being a human. And as I told you, that the creativity, learning, excellence, sophistication, parallel processing, memorization, number games, art and craft, these all are part of the human system, which unfortunately the other human-like creatures do not enjoy. They cannot even, utter simple words. For example, we use 26 letters in English. I mean, they could have used 13 letters, 10 letters. They could have used some broken, words, but nothing like even, water and food and amma and abba. They cannot say like that. They are also evolving with us only. They are there, we are there. So why this system has favored us only? Why we have only become so much intelligent? That question is still there. I would like you to find the answer.
Yes, sir. Thank you.
Over to NRC Chittoor, they have raised their hand. NRC Chittoor, PVK
Thank you,
Chittoor.
sir.
Just a minute, madam. NRC Chittoor is asked.
So NRC Chittoor, sir, we are from NRC Chittoor. Good afternoon, sir.
Yes, sir.
I have a question. You have mentioned that the penicillin invention was given to the mankind. Are we cherished or abused? Will you elaborate on this? We are confused.
About the antibiotic.
Sorry. Penicillin invention was
We
given.
have abused it or we have cherished it?
Have we cherished or abused? What does it mean?
Yeah, so we means what did I mean is that whether we have judiciously used the antibiotic, then we have cherished it, we have enjoyed it as a therapy, as a molecule, as a magic bullet. We have abused it means we have taken it, overdoses of it, we have bought it over the counter, we have fed to the animals in their ration, and now the residues of these antibiotics are coming into the water, coming into the food, and these antibiotics have exposed our system, and therefore the resistance has become rampant, not only to penicillin but all other, all other molecules, all other antibiotics. So that's the meaning. Cherishing it means judiciously using it. Abusing it means allowing the resistance to happen by misuse.
Okay, it is causing to the antibiotic resistant.
Yes.
Are we talking about antibiotic resistant?
Yes, sir.
Okay, sir. Thank you.
Over to NRC DKW.
Yes, sir. Responding, sir. So
Yes,
it's
please.
my privilege to give vote of thanks on behalf of NRC DK Government College for Women Nellore. Thank you, sir, for your wonderful and thought-provoking presentation. Sir, you have nicely explained us about the topic, process of evolution from bacteria to higher human species, perception of function and adaptation towards cognitive evolution, biotic initiation of genetic molecule, evolution, high level temperature, geothermal, prebiotic synthesis of RNA, and the complexity of nucleus and nucleoporine proteins, 34 nucleoporine proteins, natural selection, which could serve as a factor to reduce the deterioration of life. So you have given us several examples like Helicobacter pylori, virulence protein, oncoprotein KAGA, and World War. You explained about World War and penicillin chrysogenum, and as a outstretched hand of opportunity by the discovery of Alexander Fleming. Thank you, sir. We have really enjoyed your lecture. Thank you very much, sir.
Thank you so much. May I know log out, please? Thank you so much for giving me the opportunity. I am now logging out. Thanks again.
Thank you so much from all the analysis that was this stage.
Thank you for your patient listening.
Thank you, sir. Thank you, sir.
Thank you, sir.